You should now. 1, 2, 3. Hey, hey. 1, 2.
Good morning, everyone. My name is Tanya Dodgen, and I'll be your session chair today, which means I'm going to be the one holding up fingers at you when you're approaching time and helping with the slides if necessary. I'd just like to start by acknowledging the traditional owners of the Cairns area and the land and sea countries where we live and work, and show my appreciation for the time we got to hear some of their stories this morning. So without further ado, the first speaker today is Katharina Fabricius from Ames, and we'll just jump right in.
Good morning, everybody, and Thanks very much to the organizers to getting this conference together. It's a privilege to be here. So we hope the technology will work. I'm Katharina Fabricius. I grew up in Germany, and in 1986 I heard a talk about Crown-of-Thorns Starfish when I was a student and decided that's what I want to do in my life. And it was actually from Ames, and it was Russell Reichelt, many of you know, giving a guest lecture on Crown-of-Thorns, and I thought Ames is the best place in the world to study coral reefs. I really want to solve the Crown-of-Thorns issue in 1988. Came to Ames in 1988, been in many places around the world since then, but have had the privilege to work on the Great Barrier Reef and coral reefs all around the world since then. At present, we are very strongly involved in the reef restoration and adaptation program, and obviously reef restoration cannot happen without ecological underpinning. So we've been having the chance to put together a big program called, we dub it EcoWrap, to provide the ecological underpinning to do the interventions that the reef need. Before I start, I would like to acknowledge the traditional owners, the Aboriginal and Torres Strait Islanders of the land where we are working and meeting today. I acknowledge the elders past, present and emerging, and I also acknowledge the present elders having given us the consent to work on their sea country. So for those of you who are not familiar with the ARA program, Reef Restoration and Adaptation Program consists of a whole number of sub-programs that develop interventions and a number of cross-cutting sub-programs. What I'm talking about is the ecological intelligence for reef restoration. And as I said, we're dubbing it EcoWrap. The objectives of EcoWrap are to provide the underpinning. There's so many knowledge gaps. We cannot understand all of it. We cannot support reef restoration if we don't know what species are living where. We've got very little information on vital rates of corals, for example. We've got very little information on what are the limiting factors in reef recovery. So our purpose was to really develop a 10-year program to look at what is happening in the reef and how can we support reef restoration through ecological knowledge. That data is critical for the ecological models that are being developed in parallel, for the decision support, and also for the development of deployment guidance that is happening as we speak. So the Great Barrier Reef is obviously a hugely diverse ecosystem. It is as big as Italy, with 4 degrees temperature gradient almost from the north to the south, which means a projected 2.7 degrees warming is well accommodated in the reef if we are smart about it. But different regions are warming at different rates. The south, that's a very outdated graph. But if you look at the blue bars, you can see the southern area has been warming 3.5 times faster than the north. And so we've got a huge range of habitats, we've got a huge range of environmental conditions, and each reef region is different. So we've got to cater for different environmental conditions in order to decide what to do. For the EcoWrap, we've set up 12 reference reefs up and down the Great Barrier Reef, from the Torres Straits up in the north all the way down to the south. The brown reef circles demarcate the regions in turbid water environments, and the pinkish ones in clear water environments. So you're really trying to cover the whole latitudinal gradient as well as in inshore to offshore environments for the GBR to set up these reference sites. And that's how it looks. In many cases, we also needed within reef information. So each reef has got sets in the sheltered and in a wave-exposed area, in the shallow and in the deep. And so it's a very complex set, a nested design of reef plots and sites up and down the Great Barrier Reef. So we've got 88 sites, 350 plots to do the work. And those plots are really covering a very wide range of reef habitats, from algae-dominated areas to very hard coral-dominated areas, high silt environments, and clear water environments. What we're doing on those reefs is we are studying community dynamics, community vital rates of corals. We are looking at how the early life stages on the coral reefs are faring, and we're looking at cryptic species. It's not me. But we're not only looking at corals, we're also looking at species interactions such as microalgae and the role of fish in determining reef recovery rates. So there's a lot of biological, physiological and environmental studies going on at these sites. We'll see a number of these results throughout the next 3 days, and I'm just providing a quick overview of what EcoRub is all about. So Renata, who's here, has been key in developing photogrammetry tools to quantify things like structural complexity, which are a key predictor for habitat quality on reefs. If a reef gets disturbed, it typically gets flattened and is not as good a habitat for fish and reef-associated organisms. We're looking at vital rates. We think we know how fast corals are growing, but you would be surprised how few information data sets are actually available from in situ in the field in relation to the environmental drivers. We're looking at species prevalence. Where do certain species do the best? If we're deploying corals, where should they go? Which species should we choose if we've got a certain site to deal with? So there's a huge amount of spatial ecology happening, and a lot of studies by PhD students, postdocs, and scientists. Relevant is obviously we need the tools to deal with this vast amount, really terabytes of data from those photogrammetry plots. So AI is being developed to extract segmentation for colonies, identification of species, to track colonies from one year to the next, and so on, to get the vital rates and to really get a better understanding of what the dynamics are on those reefs in different places. Those tools are also became useful now in the response to this 5th mass bleaching event we're experiencing on the Great Barrier Reef as we speak. So today you will have seen the 4th global mass bleaching event has been called upon by NOAA, and the Great Barrier Reef has experienced widespread bleaching up and down the Great Barrier Reef. On the left is a plot put together by Jess Roth from CSIRO, which shows the degree heating weeks. And literally, over 60% of the reef has seen very high or extreme levels of heat stress. And the results of the aerial surveys that Ames and Kabumba have conducted will be released tomorrow. So Torres Strait and the far northern Great Barrier Reef have fared, fortunately, a little bit better. Very few reefs have bleached up there, but the northern, center, and south has been very severely hit yet again. And that's the 5th mass bleaching event since 2016. It's not surprising the reef bleached. This year has seen 1.3 degrees above pre-industrial temperature ranges, and because the temperatures are rising so rapidly, we're going to see probably annual bleaching events as of initially they said 2040, but it looks like it may be happening much earlier now. So Renata and team have done the photogrammetry plots. Here's Heron Island in the shallow water, and you see the diversity of colony responses. Some colonies are badly bleached, others are faring better, and that's exactly the information we need to extract from the reefs in the wild populations to understand and to help the reefs recover and to predict and to accelerate the adaptation to these warming temperatures. That's a brief summary from In-Modo surveys put together by Neil Cantin only yesterday. About over 90% of reefs had more than 30% bleaching in the northern. That does not include the far northern reefs, but 66% of reefs showed mortality. So it's really yet another really sad bleaching event. It's really happening, and that's why RRAP is becoming so urgent in order to understand how we can help the reef to recover. Obviously, relevance for reef restoration. We are in the process of developing tools to better quantify bleaching status, not just by in-water surveys, but also by photographic data extraction, quantifying how much a coral has bleached. We do need to understand the effects of bleaching on coral vital rates by having surveyed which corals in our plots have bleached. We go back next year to see how they fared. Has it compromised their growth rates, and, or have they survived and also come to the bleaching? Along the same plots, we are looking at fish communities, we are looking at macroalgae communities, so there's a lot of work happening here. Again, the AI tools developed to quantify fish densities. We are looking at the role of macroalgae as habitat for fish in many places. When the coral is missing, the macroalgae become the main structural element on the reefs. We are looking at the contribution of fish to reef health in terms of herbivory, corallivory, and we are looking at the role of macroalgae in diminishing the capacity of reefs of young corals to settle. Similarly, Christopoulos and others are doing a lot of work on coral recruitment, connectivity. We've been looking at juvenile densities, and I'm just showing you a few results here, which I only put together yesterday, so it'll be a little bit rough, but obviously, again, hugely important. We are moving corals, we are moving coral slicks, so understanding connectivity, natural connectivity between reefs, the drivers of coral recruitment, the environmental limitations in reef recovery are key sets of information for EcoRAP. These are the data. They're much more complex models underneath, but I decided to go for blue and red bar graphs. Overall, the densities of coral recruits on reefs are still pretty high. So we are averaging over 90 recruits per square meter, and that's up and down the GBR. But there are up to 10-fold differences between reefs. Like Keppel Islands were the worst, and 10-fold higher densities of recruitment in places further offshore. In particular, Lady Musgrave and Lizard Island have been record numbers of coral recruits in the 2 years where we had the settlement tiles out. Remarkably, to go back, the red bars are the reefs in the turbid reefs, and the blue ones are clear water reefs. And you can start seeing the fundamental effects of water quality, which I'll show you in the next few slides, in determining coral recruitment. Coral juvenile densities, these are all corals less than 5 centimeters in size. Remarkably similar patterns to the coral recruits that are arriving on the reef. So we really think we are seeing some recruitment limitation in the reefs in terms of the arrival of larvae and not later onset of survival issues. Again, the Keppel Islands had the lowest density of coral recruits. In the literature, it's commonly assumed that 6.2 recruits per square meters would sort of be the lower limit of reef recovery. Keppel's are on 8. We still need to refine those numbers, and Manu is working on those things. But at this stage, the red line here suggests where the coral recruitment may become limiting. In most places, coral recruitment is way above what the reefs would need to bounce back. But again, the 3 turbid regions have much lower juvenile densities than the clear water regions. And if you look at the ratio of clear water to turbid reefs, and again, this is a really simplistic display, but there are analysis behind it that support it, it shows that the ratio of recruits on the tiles is 3:1 between clear and turbid reefs, and the ratio of juveniles is 2.5:1 between clear and turbid reefs. So water quality really matters when it comes to coral recruitment and reef recovery through that mechanism. There's obviously coral growth and mortality as additional factors to consider. But interesting, the ratio of recruits to juveniles is about 5:1, both in clear water and turbid water reefs, which again shows that it's not later survival once the corals have settled, but it's the early settlement and post-settlement numbers that seem to be the critical bottlenecks for those reefs in the turbid environments. We also use the tiles to look at carbonate accretion. Basically, in this case, it's a fairly simplistic dry bed an assessment of how much these tiles had accreted in 2 years' time. Again, the Keppel Islands have been faring the worst, and the rates of carbonate accretion was much higher in the south than in the north. So there are some differences again in water quality. The ratio is about 1.5:1, with much higher accretion in areas of lower areas of sedimentation, and in particular the more than twofold differences in the lower levels of sediment weight, and that really makes a difference whether carbonates are accreting independently of what corals are doing. I mean, reefs are accreting through coral growth, but when the corals are missing, they still are being eroded through wave erosion and so on. So we need to know how much is actually happening on those 80% of reef area that are not covered by corals, and what's going on here. So again, a water quality issue here in terms of reef accretion, and again, turbid Keppel Islands are unfortunately struggling a bit in keeping up the maintenance. So again, I mean, the relevance for REP is pretty obvious. I'm just throwing it out to re-emphasize it. What does it all mean? The reefs are facing incredible pressure. Now we've heard a good summary for Uncle Bob, sort of pointing out the critical time we're in. We're really in a critical decade after hundreds and thousands of years of relative stability. We're changing the environment in an unprecedented rate. The reef disturbances are becoming more frequent. And so here, the little schematic I put down here is much better done by the modelers, but what it tends to show is we did have natural disturbances in the past, and there are capacities of the reefs to recover until now. If the frequencies of disturbances are increasing as it does now, through bleaching, through more intense cyclones, through more intense floods that bring more sediments into the water, because the warmer air holds more moisture, therefore rainfall intensity becomes more extreme. We are facing a time where reefs are becoming more often disturbed. So REP stuff will hopefully be able to, we can't prevent the natural disturbances, but we hopefully can help the reef to adapt to the warming temperatures a little bit, so we are not facing a wipeout, but some sort of dynamics of some semblance of coral reefs as we've known it in the past. So in conclusion, I believe at present all the data we've got are that most coral reefs are still quite resilient, but there are exceptions. The Keppels are one example where the reefs seem to be struggling a lot. Keppels have got high sedimentation, got the Fitzroy River to contend with. But many other places show very high recruitment, high carbonate accretion rates, and coral reefs are naturally dynamic. They are able to recover from natural disturbances. So the main problem now is the frequency of disturbance, but the dynamics are such that disturbances are fast, but then it takes years for reefs to recover, probably 10 to 30 years. So assembling those life history data takes time. EcoRep started 3.5 years ago, and we've achieved an enormous amount of time, but it has been designed as a 10 years program. So we are only just starting getting those information of vital rates, how fast the coral's going, or the reproduction rates, and so on. So it's early days in terms of showing you results, but there will be about 10 talks for the next 3 days showing you some of those highlights of the results that I've summarized here. We can now answer more questions about heat tolerance, about we're working on issues like connectivity, But nevertheless, it is a critical decade. Without climate mitigation action, the reefs will have very little hope. But the water quality links I've shown you here also shows very strongly that traditional reef management action is still immensely important, and probably more important than ever, because we know it works. We know water quality leads to better reef resilience. We know fish are really important in allowing reefs to recover. So we don't replace the traditional management with some fancy respiration. We need more of those things to really support the reef into the future. Yeah, and that's a big thank you for you to listen. The work could not have been done without those 45 people who are the core EcoRUP members. I highlight in bold the people who may be talking in this conference. And I would like to really thank the Arab managing entity, the GBIF, the conference organizers, and all the partners who have been supporting this Arab program in this journey for the first 3.5 years. Thank you.
All right, we have about 5 minutes for questions, and we'll be doing a dual approach. So we have a Slido assistant in the corner here. Yvette will be assisting throughout the day with Slido online. And we ask so that the online audience can hear that you line up for the microphones if you're here in person and would like to ask a question. Thank you. She can repeat your question if that's easy.
You have talked a lot about the relationship with water quality and the sediment and turbid waters, but what about the nutrients? Is any indication as well as part of your research that is showing that relationship with high nutrient levels?
Yes, so there's a lot of research going on the effects of water quality on the Great Barrier Reef, and I've been involved in that, as you know. Water quality is difficult to tease apart which factor in particular is affecting things, because in many cases sedimentation and nutrients are highly confounded. So yes, we know that dissolved inorganic nutrients make algae grow faster, sediments affect corals more. So in many cases there are different pathways towards different effects. High nutrient runoff from cane farming in particular is of key concern for us. High sediment runoff from, in particular, grazing areas is of high concern. So there are regional differences, and in this sweeping talk, I can't really go into details, but nutrients and sediments are both a key importance for the coral reefs. That's work in progress, and because we've got these photogrammetry plots that give us species compositions, we've also got the species composition in juvenile densities on the tiles. On the early recruits, it's impossible to tell them to do anything greater than families. But yes, these relationships, we're just calling for postdoc now to look at those relationships in detail, because it's a beautiful data set.
We'll just do one last question in person and then one online if there are any online. I'll repeat the question on the microphone. What? Sorry? Yep.
Is there any way to tell the difference from the EcoRUP data on, say, for example, the site that's showing poor recovery, whether it's actual larvae in the water that larval supply is low, or whether the larvae are there, but the substrate conditions in the river just poor settlement due to the, if you can't actually measure the quantity of larvae in the water, all you're looking at is the settlement or the recovery. How can you actually work out what the recovery is if it's supply or sediment conditions?
So to repeat the question, is it possible to use EcoRap data to tell whether recruitment is limited by larval supply in the water column or whether it's limited by available settlement area and conditions?
Thanks, really good question. And probably a third of the EcoRap program looks at the early life histories, and that's led by Christopoulos. There are whole projects looking at connectivity, larval supply issues, and trying to tease that apart. But we haven't got the answers yet in this particular data set. So, but it's a very important question, and we are working on that. Thanks.
Do we have any questions online? Okay, thank you very much for opening this session, Katharina. The next speaker is Adriana, and just a little update that the subsequent talk by Alexandra is cancelled. Oh, you don't look like Adriana. Is by Juan. We'll let him take it away with the next talk.
Hello, everyone. We'll get to the point of my name in a second. So I wanted to start by acknowledging the traditional owners of the land and sea country where we work and where a lot of the EcoRap and RAP data is collected from, and recognize the elders past, present, and emerging. And I wanted to make a reflection after hearing Bob talk this morning. It came to my mind that we've been thinking and recognizing the traditional owners as the original scientists of the land and sea countries where we are. And in the case of the Great Barrier Reef, even though corals have been around Australia for about 500 million years, the current Great Barrier Reef, the one that is World Heritage, has only been there for 10,000 years after the last glacier age, which basically means the traditional owners of Australia have actually been the custodians of this ecosystem from its birth, for the whole time it has existed. And I wonder if there is any other example of an existing culture that has actually looked after an ecosystem that exists now and that they have looked after it from the time it was born. So I think that's really interesting. Now, going to the name, I'm not Adriana. We come from the same country, if that helps. But we have been collaborating in this space. My name is Juan Ortiz. I work at AIMS. I come from Venezuela. I started snorkeling and diving in Venezuela when I was about 12 years old in some shore reefs or a beautiful national park that is there. And I remember clearly how in the back reef on some of those little cays, there were all these cropora branches, some dead, some alive, and so many lobsters in between the branches. I remember snorkeling and thinking, this looks like a supermarket. You just dug down and you cannot see which one is the biggest of the lobsters because they all come out and show the antenna and all that. And then I went to uni to study ecology. I had not decided to do marine biology yet. And I started learning that the Caribbean reefs were quite degraded. A lot of that information from the Central and North Caribbean, and I kept going to the reef, and I was like, this is weird. I don't see this. I mean, reef still looks great here. And sure enough, a few years into uni, I went back to that beautiful place, and it pretty much was no longer there. And it hit. And then I went to the Northern Caribbean and said, well, this has been happening for quite a while. That's when I decided to do coral reef ecology as opposed to ecology in general. And then I came here to Australia, and I feel a bit like now we are in the same scenario. The Great Barrier Reef, as Katharina was saying, still has great recovery. We have great recruitment, but we have a big threat. And if we don't do something in time, we may repeat the experience some of us had already from the Caribbean. So let's hope we can help a bit and avoid that from happening. So I'm going to be presenting the work that we've been doing in relation to ecological heuristics to decide on site selection for survivorship and growth of deployed corals. And it's a work that has been led by Adriana, who's also from Venezuela and is in Palau at the moment spawning corals. I spoke to her last night. One of the species behaving really well, the other one not so much. And the internet connection wasn't good enough for her to be presenting remotely. The title of the talk that is in the abstract is the one in little font that you probably can't read. I made the bigger one just to be a little bit more entertaining because it's all about trying to avoid the little corals that we love so much and look after and try to make special from dying as soon as we put them out in the reef. So which one is the one that? Not that one? No, this one. Yeah. So the challenge, right? We are trying to influence reef resilience in a scale that has never been attempted before quite yet. We are trying to produce millions and millions of baby corals through different processes, whether it's bring them from some places or growing them in different places, but numbers that are quite huge. We are trying to find ways to make those corals a bit more tolerant by harnessing the information we have about their thermal variability through either symbionts or the genetics of the host, et cetera, et cetera. Already a big challenge. And then we have been thinking a lot about finding the right place to put them. So because we have to upscale, we need to get enough of it moving around. The spread of them is enhanced by connectivity, and we can be really smart and find the perfect place where we will get the most bang for our money. However, there is one critical point. It doesn't matter how well we can identify the perfect site for them to spread. If the corals we put there don't grow, survive, and get to be reproductive, we won't get it, even if it was the perfect site for a spread, right? And in that sense, we know from the literature and the knowledge in ecology that small corals are challenged. It's a bit like the National Geographic documentaries of Africa. It's a struggle for quite a few months and years for them when they get out. They have very low survivorship when they are little, and there are many reasons why they may not do so well. We also know that we can't really protect them that much. It's millions that we want to put out. We can't have a mom looking after them, each of them scaring everything away and making sure that everything is perfect for them. That's never going to happen. So that means that we have to find ways to use what we know to identify places where they have the best chance of growing, surviving, and becoming reproductive. And in that sense, we have started working. Those letters are really small in that screen from here. So we have started working on these rules and using the available information to start to develop a criteria under which you can rank sites based on their biotic and abiotic characteristics to try and maximize reef, the survivorship and growth of these corals that we are deploying. So ultimately, what we are developing here is a set of rules that allow you to rank a set of sites you are considering for deployment as a function of how likely it will be that the corals that you put there may grow, survive, and reproduce. And in that sense, we have developed a framework that is now already being discussed and used within our RAP that consider 3 different criteria. The first one has to do with the difference in environmental conditions between the place where the corals, the source corals are from, and the receiving sites where you are planning to put them. And we know that, as Katharina was saying before, the Great Barrier Reef and reefs in general are very complex, and there are very different environmental conditions in different parts of the reefs, especially, and also within a reef. And we also know that it's quite likely that if you bring corals to an area where the environmental conditions are fundamentally different to the ones where they come from, there may be challenges for them to thrive in that environment. So the first part of the criteria has to do with how similar or different those two source and receiving sites are. Then the second 2 parts of the criteria have to do specifically with the receiving site, and the first part had to do with the abiotic characteristics in that receiving site, and the second part it has to do with the biotic characteristics in that receiving site. And then this set of criteria are standardized and put into a format that allows to provide indices for each of these three aspects, and then overall indices for the combination of that when you want to rank all of the sites. So the principles here is that we use a combination of available information, new data, and mechanistic understanding of, current mechanistic understanding of the ecology and biology of reefs to come up with these criteria, which are still a work in process and will continue to be a work in process even after it's published, because we always get more information and better information. The idea is that the criteria can be evaluated through different approaches depending on the scale you are looking at. So if we start a bit with the first, the difference in environmental conditions between the donor site or reef and the receiving reef. And in that we have a set of criteria that compare whether the species that you are bringing already exist in the existing environment, whether the temperature regime is comparable between the two, whether the other environmental conditions that are well known are fundamentally different between the two, and when possible, we have provided thresholds and quantitative approaches to standardize these differences and decide whether that's 2 minutes left or 2? Yeah, all right. So, so when possible, we have decided and found those thresholds to be able to get to those. Importantly, the principle here is that when you apply these criteria, any of them, you get an index that the higher the number, the better the site. But in some cases, you have a no deploy output, where it says just doesn't matter, don't consider it, don't put it anywhere, just don't deploy there. When we then go to the abiotic characteristics of the site, we consider what we know about what little baby corals or small corals need to be able to survive. And we consider the slope, the rugosity, the keep you factor, which is still debated whether we'll keep in there or not, has to do with structural complexity, the wave exposure, and we use the available information we have to create a standardization that allows you to then get the values from the index and compare the sites. Now, these two aspects that we just described, the first two, most of it can actually be evaluated via desktop exercises because there exist spatial layers and spatial maps that are available that allow you to explore almost any site around the Great Barrier Reef with some limitations without having to go to the site. And this is important for what I'll say in a minute, in the pretty much last minute. So finally, in the biotic components, this includes how much coral is already there, how many coral, what's the density of corallivores in there, what are the conditions in terms of biological interactions that may prevent or maximize the survivorship or growth of these corals. Importantly, though, this part of the criteria we can't really evaluate from desktop exercises because first, the information is not available, but second, is dynamic. So even if we had spatial layers, they wouldn't be constants over time. So for this part of the final part of the evaluation, you need to go to the site and evaluate the criteria that is presented there. And that, over time, that could be done with traditional measures, and over time, more autonomous approaches may be developed that allow us to do that. However, there is only going to be a challenge because you can only go to so many sites. And there may be, in many situations, many more sites that are considered than the ones you can visit before deployment. So in that sense, we have developed the approach in a hierarchical way where the first two criteria can be evaluated first and filtering of bad sites can be ruled out. And then a smaller number of sites are the ones that are evaluated for the site visits and the biological conditions, which makes it more logistically manageable. And even in the worst scenario where after the filtering, you still have more sites than what you can visit, you still would have a ranking, so you can start visiting them from the best out of the first two rankings all the way down, and basically stop when you run out of time and still get important information. So the approach has now been prepared as a paper and it's been submitted for review internally in the internal process in IRAP and AIMS, and it's going to be submitted out very soon. And it's been already used, evaluated, and improved through different sub-programs in IRAP, including CAD, modeling and decision support, and their 2025 deployment guidance system project. And I think that's the end.
Great. Is this coming through? All right. I'll just echo questions from the audience up here so that we don't have to scramble out of our seats. Do we have any online questions? Nope. Any from the audience? Yes, over there.
Has anybody done any study on the survival in a difficult environment when deployed to the Acanthaster farmer fish territories? Because I know there's evidence that that's where they recruit better. Is there any evidence that, I mean, like, because they protect them from crown-of-thorns and parrotfish and everything else? That's what we're finding, is that if we have a problem site, if we locate them critically in a farmer fish territory, they do so much better.
So the question is there any research in this area about intentionally targeting farmer fish territories for larval sediment as for any outplanting, as they've observed success in those areas due to the farmer fish removing Crown-of-Thorns starfish?
So I know there is some research in this space. We haven't considered or included in the criteria at the moment, which we can reevaluate because, as I said, this will continue to be growing. There are some challenges in that space because part of the approaches that are being considered is to be able to upscale the deployment in a site. And the issue with being so restricted to such a small area within the site will require really guided deployment that will limit the scale at which or numbers that you can provide. So even if we were to bring it into the criteria, which we eventually could, it would probably be for very specific scenarios where you are focusing in a particular reef where you really want to maximize for a reason, and then you may go down that path. But for the broad application of the upscaling, I think it would be very tricky to implement. But we haven't explored it, so this is just off the top of my head.
Yep, question from online. Are the criteria fully applicable globally?
I think that question will depend on the person you ask. We thought of them and decided them with the hope that they would. And also the actual package, which is a dynamic Excel spreadsheet, provides a lot of functionality in the sense that you can define the objective for which you are trying to apply the criteria. You also can change the weighting of the different criteria, which is something that may be quite relevant when applied in different parts of the world. And we did try to incorporate the main mechanisms that are general and apply everywhere, but it's still yet to be tested whether that's the case. So once it's released and people start playing and thinking about it, that's when we really know how well it works. I'm sure it won't work everywhere the same way, but hopefully it can work in a broad enough set of conditions.
All right, thank you very much, Juan. Welcome to the next speaker, Mariana.
Hello. Thanks for coming. Sorry, I thought there was somebody else before. So I'll just start by mentioning this is the work from many people. I'm just here the one presenting, but there was a lot of people in the water and then out of the water processing the data. And I'd like to start by acknowledging the traditional custodians of the sea country where these data were collected and where we meet today. So as you all know, corals have a symbiotic relationship with algae, and when they get stressed, the symbiotic relationship is broken and they expel the algae from the coral, so that's when we see the white skeleton underneath. The main reason for these, or the most usual reason, is when temperatures exceed what the corals are used to. And this is exacerbated by high light intensity. But there are other factors that affect how corals bleach, or the proportion of corals that bleach. So depth is one of the factors that has been looked at more closely. As you go down the water column, you would expect slightly colder temperature and less light intensity, so it's thought that it should ameliorate the bleaching. And there's lots of studies on this, but there's studies presenting both patterns, so where the bleaching decreases with depth and also where it increases. And it's very taxa dependent as well. Another factor, important factor here is water velocity. So as you get more faster water flow, the mass transfer between the toxic byproducts of the bleaching and the water column is easier. So it's thought that it should be less proportion of colonies should bleach when there's high water flow. And again, there's been studies looking at this, and the patterns have been shown in both directions as well. Taxa, I think there's been a lot of effort here. So there's the winners and the losers, the constant winners and the constant losers, and then lots of variation in between. And these changes within a site, within a species, and also between the different studies and the different bleaching events. And the last one that I'm going to talk about today is colony size. So most of the effort has been looked at comparing what the juveniles are experiencing and the adults are experiencing, and less so kind of the range, once they're already adult, how does colony size affect bleaching? But in general, recruits or juveniles bleach less than the adults. So as I mentioned, there's lots of effort that has already been done, and we're still finding high variation. One of the limiting factors here is the time spent underwater. So obviously when you're down there, you can only record so much data. So photogrammetry can help us get capture the patterns on a much broader spatial scale, and then do all the time-consuming bits kind of the water when you're not so time-limited. So that's what we did in April 2022. So this was a mild bleaching event in the central Great Barrier Reef. And Katharina already showed us kind of how the different sites were laid out, but I'll go through it a little bit again. So we had 4 different reefs. The Palm Islands, kind of our inshore reef, and then we had Chicken, Davis, and Little Broadhurst in the offshore reefs. And then each reef had sites all along the periphery at 5-meter depth. And then the Palms and Davis Reef had also sites at 12-meter depth. Each site had the 4 plots that Katharina showed, and in total we had 104 plots. So this is an example of one of the orthomosaics and a little zoom in of the colonies. So once we were out of the water, we traced the colonies so we could get colony size estimates, and then we labeled them according to the bleaching. So there was kind of no bleaching, minor bleaching, from up to 50% of the colony was bleached. Then major bleaching, which was from 51 to 95, and then anything above that, including whole colony mortality, was labeled as severely bleached. And we looked at a range of different factors, some of the ones I mentioned. We also looked at other factors that I won't have time to go through them today. So this is kind of a subset of what we found. So the first thing that was surprising, the degree heating weeks. So for those of you who might not know this, it's a metric of the bleaching stress. So basically, the longer and the higher the temperature relative to the temperature that the corals are routinely exposed to during summer, the higher the degree heating weeks. So most of the studies find a really steep curve there. And we did find a positive relationship. It was not very weak, but if you can see I don't know if the numbers are big enough, but the variation there is not too high. So it goes from 4 to 5. So basically all of our sites were experiencing a very similar bleaching or thermal stress. And then in terms of water velocity, our patterns differed between the inshore and offshore reefs. So in the inshores, we saw that decline in bleaching susceptibility with water velocity, which was the one we were expecting, but not so much in the offshore. And the offshore did tend to bleach a lot more than the inshore, which kind of makes sense because it should have higher light intensity due to less turbid water. And one of the things here to consider is that the water velocity we had to extract from maps, so model wave data, and summarized through time. So it might not really reflect what the corals were experiencing at the time of the bleaching event. And here I just showed any bleaching, so I didn't divide it by different categories because they were all behaving really similarly. In terms of taxa, we got some of what we expected. So for example, Ceratopora hystrix, Stylophora pistillata, they're usually on the losers category, whereas the pride is massive. They don't tend to bleach as much. The one that was kind of surprising was the Acropora table, which didn't really bleach that much in our study. And this was in contrast to what Paul Marshall and Andrew Baird found in the Palm Islands on the 1999 whatever, bleaching event. So that was the category of the most severe taxa, severely bleached taxa. So I'm not sure what's going on here, but observer bias is not likely to affect the ranking, at least, because it was just one person. And in terms of colony, the effect of colony size, you can see there's positive relationships, no relationships, and negative relationships, depending on taxa. But if we looked a little bit more closely to the different bleaching categories, so minor bleaching usually increased with colony size, whereas severely bleached corals decrease with increasing colony size. So I think this suggests that the larger adults are a bit less susceptible, and they were kind of on the way to showing the effects of the stress, whereas the little ones had already gone through it and they were already on the severely bleached categories. In terms of depth, there were various relationships depending on the taxa, but the signal wasn't super strong. And this kind of makes sense because we were looking at 5 to 12 meters depth, so the range wasn't as big as some of the other studies. And just to wrap up, photogrammetry was a very useful tool for us because, first of all, it allowed us to target different taxa that we were not initially planning, and it obviously helps with getting the colony size estimates. And from here, the next step would be to look at how the bleached corals differed in terms of growth and survival with the non-bleached corals. So that's probably the next step where this data set is going to go. And we found lots of variation in bleaching. We obviously didn't look at the symbiodinium identities or the genetic component, which I think Zoe Messier from Cynthia's lab was going to be looking at. And just to put this in perspective, the results I showed here, there are kind of like size refuge and some taxa that didn't show susceptibility to thermal stress. But we were on the yellow, from that map, the yellow bit is where this bleaching event was at. This is the map for this year. So it's not looking as good this time around. And this information will hopefully help when we decide like if which taxa and where to do any of the interventions. This information might help some of those decisions. And with that, thanks to everyone who helped along the way.
Thank you very much. Do you have any questions from the audience? Yes? Yeah, if you'd like.
Can you talk a bit about how we can use that relationship between solaris and bleaching susceptibility in restoration and management? How would that sort of change your decisions, knowing that relationship with it? And I think because it's different in different species, makes it very different.
Yeah.
So the question is, how will you use this knowledge about the relationship between bleaching and size tolerances to inform how can that be used in future management or restoration?
Yeah, that's a great question. I think, so some of the talk is at what size should you deploy the the juveniles or the recruits. And obviously, the longer you keep them in the tanks, it costs a lot of money, and also you might be getting some effects of the tank or just being held in captivity. So ideally, you want to deploy them as fast as possible. But for some taxa that showed that the smallest ones are much more susceptible than the larger ones, that might be a compromise where you might target some species to grow a little bit more in the aquaria before you deploy them. But yeah, I think there's a lot of things to play. Yeah, it would be pretty expensive. I agree.
And what drives that correlation between size and reef?
Yeah, that's a great question. If you have any ideas, we'll love to hear about them. I think for in the opposite, yeah, sorry.
Old people bleach their hair. But the bigger of young people and the bigger of young corals is obviously part of it. And what in terms of bigger, I don't think I need that. Okay.
It's for that we have lots of people watching online, so they can't hear unless it's
Okay.
in the microphone.
So the vigor of youth, I mean, it's obvious to me, but with disease, we find that the small corals succumb much worse to disease than the large ones in our sites. And my question was, is it a factor of size? I mean, if you break up a coral of the same genotype into different sizes, would the young ones be more resilient in the initial stages, and would the old ones be more resilient if the bleaching continues and continues and continues? Has anybody done anything like that, looking at the same symbionts and genotypes, and then you break it up and see what happens?
So like having the same colony and then seeing if you break it into different sizes, does the susceptibility change? Yeah, I'm not aware of anything.
It might not be age, it might be size.
Yeah.
Final question from the audience.
Hi, so with regard to the size effect that you found, I thought, maybe I misheard, but I thought you were alluding to the fact that the size effect could be confounded by past experience. In other words, the larger colonies have been through more, and so they're carrying that kind of latent effect that the small colonies didn't have.
Yeah, it is possible that they've experienced, like they're the survivors of previous bleaching events, and then they're less susceptible, but I think it's all kind of speculation at this point, yeah.
Right, thank you very much.
Thank you.
Sounds like there's a lot of interesting ways forward in that space. Next speaker is Pete Mumby. Thank you.
Good morning, everybody. And whilst we're all sharing, I think I got into marine biology because I watched the movie Jaws when I was 7 years old. And I discovered that you could have a job doing this marine biology stuff. And then years later, someone said to me, if you're a marine biologist, you'll never do a proper day's work in your life. That's totally untrue, as we all know. But we all have a good time. Anyway, I'd like to talk about coral reef reproductive success or failure. And this is work that's been led, it's a collaboration between UQ and CSIRO primarily, and it's been led by the postdoc Jared Ricardo, who does a fabulous job. And we're all aware that if you've got a healthy reef system, you'd expect there to be vibrant reproductive success. And the concern that many of us have is that as reefs degrade, corals become farther and farther apart, and this might mean it's less likely that they achieve successful fertilization. And the reason for this is that there's a strongly nonlinear relationship between the fertilization success of eggs on the y-axis and sperm concentration on the x-axis. And so any process that dilutes sperm, such as corals being farther and farther apart, means you're much less likely to achieve reproductive success. And this is obviously a question that might influence how you would output corals. You want to make sure that they're outplanted at distances that mean they're close enough that you can achieve some successful natural reproduction. But it's a really difficult thing to study because if you do it in the field, there are so many confounding influences. You can have other corals that will hybridize with the corals that you're interested in and so forth. And so the approach that we've taken is to work in isolated lagoons where we can control the process. We can transplant colonies from out on the reef into the lagoon we can control the spacing, and we can test for any confounding effects and hopefully reject them. And so we've been doing that both at One Tree Lagoon, which is fantastic because as the tide falls, it isolates the system, and also at some sites in Palau, Micronesia, where we get another shot at doing spawning experiments later in the year. And the sort of general setup is we create these beautiful little condos for corals, and any coral would be very happy to live there for a short period. And you have the egg bundle collector above it, and on the top of that bundle collector, we have no, we don't. Okay, on the top of that bundle collector, we have this little pot. It's about 15 centimeters high, and the walls are made of plankton mesh. So once we seal it and it's collected the egg bundles, the eggs are forced to stay in the pot, but the sperm can wash in and out so that those eggs can be fertilized. And you can see in that panel on the top right, this is in the daytime, just to give you an idea of what it looks like, that this thing will float on the surface. We attach light sticks so that we can track them at night. We'll talk a little bit in a moment about this strange green stuff. And then we can follow these floaters for at least an hour at night once they've spawned to see what kind of fertilization success will have occurred. We bring them back to the lab and quantify success. So the first experiment we did was at One Tree, and it was a really small-scale experiment. We just had 8 colonies of Cropera tenuis. We separated them by 2 meters, which is a pretty reasonable distance. And we asked, would you see any fertilization success? And to be honest, I was amazed if we would see anything. Bear in mind that when those bundles are released, you have to wait at least 15 to 20 minutes before those eggs are ready to be fertilized. And that's quite a long period of time for those eggs and that sperm to go in different directions, especially when you're starting with such a tiny pot of corals. What we actually found was sort of close to that. We did find success. There was about 4% on average fertilization success in these corals. So this is fertilization success on the y-axis. And to be honest, I was even surprised that we got anything happening. We then, on that same night, did it with a second species. This is Acropora digitifera. Now we intensified the spacing from 2 meters to 1 meter. We had more colonies, and we got slightly higher fertilization success. We then did it in Palau with another species. This is Acropora hyacinthus, 1 meter spacing again. Again, a pretty small patch of colonies, of 20 colonies, and we see a slight increase again. So what does this tell us? Well, it tells us that if you have a really tiny patch of corals that you've restored, remarkably, they do actually contribute a small amount towards the population through fertilization and the generation of larvae. But of course, you're getting pretty small fertilization success. And the real value of these experiments was to help us to parameterize a model that we could generalize. So the following year, we did a more extensive experiment. And this was done in Palau. And if you look carefully in that top left panel, we have a dense patch of colonies that are at the density that we find on the reef. This is Acropora hyacinthus again. And then we have a series of colonies on their condos at different distances. So at 0.5 meter, 3 meters, 10 meters, and 30 meters in different directions. And again, the idea was if we have a spawning event and you've got this central high density patch of colonies, what's the likelihood that these colonies that are farther away are going to achieve fertilization success as we get further from the main center of mass? And I'm not going to show you the results yet. I mean, obviously, because it's not going to let me do anything yet. But anyway, yes, we'll move forward. So the reason we did this was, again, to parameterize a model. And Jared's done a great job here, taking a model that was originally created by Teo and Pete Todd in Singapore, and then extending it. So this is parameterized, in this case, for smaller Cropera. And we spent considerable time, along with one of the PhD student, Liz Brackeri's work, looking at fertilization dynamics and modeling the dispersal of sperm and eggs. And so when you get this model, you orientate the corals in some kind of grid on your model. You then track what happens with individual corals. They release their bundles of eggs and sperm. They hit the surface, break apart. they then start to disperse. The eggs are floating, the sperm are being able to mix in 3 dimensions through the water column. There can be advection and diffusion. And then there's the fertilization kinetics, and that will vary according to sperm concentration, egg size, and so forth. There's quite a lot of complexity there. And to try and get a handle on what the sperm dynamics were, we were releasing these harmless green dyes at the same kind of tidal conditions during the daytime, of course. And then you have a drone sitting above you, which allows you to trace how that sperm is diffusing over the seascape over the next sort of 20 minutes or so. We can then use that to create the model. And this is Jared's funky little model in R, which you can see a little simulation there. Well done, Jared. In this case, it predicted 8% success. Very, very nice. So what do we see when we do that? We see generally fertilization success on the y-axis again. The gray bar is the observed data for these 3 species and these small-scale studies. And the blue circles are some of the model predictions, depending on how you're working with that model. And you can get a variety of outcomes from that model, but in general, they show the same sorts of patterns and fairly low success under these circumstances. And you can do various sensitivity analyses that I won't describe. And then you can try to generalize, and this is the key thing. What do you learn from this? So if we look generally then, on our y-axis is the size of the colony. Obviously, bigger colonies, on average, will tend to be more successful. They release more gametes. And on this axis, we've got the spacing, the distance between colonies. And you would expect the highest fertilization success with big colonies that are close together, which is the upper left-hand corner, which is exactly what you would predict. And it tails off as you get further away. And this was our goal, to have something that we've now based on real empirical studies that we can start asking questions regarding how does spacing affect reproductive potential. But we also wanted to test this out, and I've just returned from another experiment in Palau. This time we did it not with an experimental manipulation in the lagoon. We wanted to know what happens actually on the reef itself. So we're going to one of our study sites here looking at Acropora hyacinthus, and we're looking at corals of different distances, different separations. And these are some pretty high density corals. So here's the experiment in action. We've got the collectors on top of the corals. They're at different separations, and this is what we find. So again, the y-axis is fertilization success. The x-axis is the nearest neighbor from your colony to another colony that's of the same species that's spawning that night. And we did this for 2 successive nights. The first night was really calm, perfect spawning conditions. The second night was very windy, and everything changed. The success rates plummeted when we had windy conditions. And this is actually the topic of Liz Bucheri's PhD thesis. But that's what we saw out there at night. But the overall trend that you see is what you would expect. As you separate these colonies, fertilization success declines. Interestingly, the models predict that we would have something like a 20% or lower success once you separate them by around 6, 7, 8 meters, that kind of distance. And that is what we're seeing on the reef itself. So I think models will never be a perfect match to data, but it gives us some sense that, at least for acroporids, that there are some of these critical spacings that might be important. So what does this tell us? First of all, I think that coral separations of 8 meters or more are going to tend to lead to low fertilization success in reproductive events naturally. Once those corals get separated by at least 12 meters, I think we might expect very low fertilization success, at least in the acroporids we've worked with so far. And I think that this can help direct some of the activities. One obvious implication is if you were able to go and assess the density of colonies on a reef and find that they're at lower density than this, then perhaps one goal of restoration is just to add enough density to reconnect that population and switch the reproduction back on again, rather than allowing it to fail. But this needs to be done with more corals. And lastly, I'd like to acknowledge all of our partners and collaborators, in particular the Palau Coral Reef Centre. Thank you.
Great stuff, Pete. And I saw my name there, Adriana, in the acknowledgement, so that's good. My other name.
Well, you're everywhere, man.
Right. So I think this is very critical for our RAP in terms of the density at which we deploy or our RAP deploys. That's really useful. But it can be, in my view, a bit worrisome as well, because one of the objectives of what we are trying to do is to spread a particular genetic configuration that we are putting in a dense patch. And then if we just let them spawn and go through connectivity, we know that the proportion of them that are going to survive is small and they're going to go everywhere. And in a space, in a reef that is getting more and more degraded, the likelihood that they're going to happen to land and grow in a place where they are going to be close by other corn-specific is probably small and getting smaller over time. So I wonder if these results also suggest perhaps the potential of a rethink of how we follow up once we deploy and whether we should be perhaps collecting that output of a dense space and put it elsewhere in a dense space as well, as opposed to be counting on connectivity.
Yeah, that's a really good point. And I think, it clearly argues that, and this isn't going to surprise anybody, and it's not a new thing to say, that restoring at low densities is going to be probably a waste of time from a reproduction standpoint. It might restore some short-term ecological functions, but it won't switch the ecosystem back on again. How we deal with these problems of trying to maintain the sort of genetic composition of corals that are outplanting and how that gets diluted is a hugely complex question that I can't answer in a few minutes, of course. And whether that argues for trying to create high-density patches of equally thermally tolerant corals that go out there, rather than sort of spreading them out, you have them in concentrated patches so that they do at least interact with one another, would make sense if that's what you're trying to do. But one of the real problems we have here is that hardly anybody assesses the density of coral species. And so even just trying to get data on the density of individual coral species across a reef is difficult. There's very few data sets that have done that. We work on coral cover, life forms, and so there's a lot we need to know to be able to operationalize this more effectively.
I think we only have time for one more question, sorry.
Thanks, great talk. I'm just wondering, for the studies where you translocated the coral broodstock, how synchronised was the actual spawning on each night, and would you expect fertilisation rates to increase if there was 100% of the broodstock that spawned? Yeah,
good question. So interestingly, the typical thing that we see is that on one night you get a dribble of a few colonies, and the next night they all go. So we did have high synchrony in that regard. When we did it in the field just recently, again, the first night was a combination, and we recorded this, of course, a combination of a bit more than a dribble to moderate spawning of colonies on that first night during these perfect conditions. And then the second night was when everything went very strongly. And although a couple have gone completely only on the first night, most went both nights and more strongly on the second night. And yet that's when we had the poorer outcome. For the same corals had much lower fertilization success when it was windy. So, yeah. Okay.
Thank you very much. The next talk is Josie.
So good morning. I'm going to be talking about our work on COTS feeding rates, which is a CSIP-funded project. So as you're all aware, it is the feeding behavior of Crown-of-Thorns starfish which make them famously problematic for our reefs. Feeding by COTS is considered to be one of the major and acute threats that undermines reef resilience on the GBR. And as a result of their devastating and costly impacts to coral reefs, COTS have gained major research interest. However, since the 1990s, very little work has been done. This isn't working. There we go. Very little work has been done on the feeding behavior itself, which has left major knowledge gaps. Having accurate estimations of COTS feeding rates, for example, is essential to understanding their impacts on coral assemblages, and understanding that is essential for effective management of coral reefs. Feeding rate data can also provide us with additional insight into starfish nutritional condition during the course of an outbreak, which is also currently lacking. So with that in mind, we designed a study with the objectives as follows. So firstly, to use advances in technology to establish highly resolved feeding rates, and a key aspect of this would be trying to obtain coral consumption rates at both a 2D and 3D surface area resolution. Secondly, to identify which factors were really driving variation in COTS feeding rates with our large-scale survey design. And lastly, a really important aspect of this study was going to be sharing our new and improved estimates of daily feeding rate with modelers and managers to enable the best possible ecological projections and decision-making. So in order to enable this to be a really large-scale feeding study, which encompassed a huge number of COTS, we used the SALAD survey technique, which is capable of surveying over large distances. And you'll hear a bit more about this technique with Morgan's speech later. But what you need to know is that during surveys, for every starfish that we encountered, we took measurements of the individual itself, and then recorded all of the fresh feeding scars in the vicinity to the starfish, and identified them to genus, and also measured them. So for consistency reasons, we considered a fresh scar to be one where the skeleton was lighter than a B2 on a coral watch card, as you can see there. So all scars that we encountered which were darker than a B2 were ignored during surveys. So we previously conducted an exercise where we photographed the aging of COTS scars over 14 days and quantified the point at which scars surpass a hue-based threshold equivalent to that B2. And this was found to be 6 days in the summer months and 10 days in the winter months for a whole series of scars that we did this process for. And this was going to be really important for us later when we were trying to establish daily feeding rates, because we could assume that when we encountered a series of fresh scars on a SALAD survey, all of the scars that we were calling fresh would have been consumed within that time period. So these surveys were conducted over 24 reefs in 5 distinct regions of the GBR, from Townsville in the south to Cape Grenville in the north, over a total of 208 SALAD surveys. A really important aspect of this survey design was that we wanted to be surveying reefs over a whole gradient of latitudes, population density, and coral cover, so that later we could really try to investigate trends in feeding rates. So from these surveys, we were able to collect a huge amount of data on scar measurements. However, as I mentioned at the beginning of the talk, we wanted our feeding data to be a resolution beyond just linear measurements of scars. We saw value in collecting data on 2D planar area of feeding by COTS, and then also the 3D surface area of feeding as well. So photogrammetry presented an opportunity to collect this data. However, it was going to be too time-consuming for us to photogrammetry every single scar that we came across on our SALAD surveys. So what we did instead was we went ahead with the rapid field measurements of scar and colony, and then later used conversion metrics to convert these to 2D planar area and 3D surface area. So unfortunately, there weren't any conversion metrics which were suitable for what we wanted to do at a morphotype-specific level. So we conducted a side project with the University of Sydney, where we modelled the relationship between field measurement of colony and associated surface areas. So what you can see here is an example of one of these models we established for the genus Poecilopora, and this model was constructed from the photogrammetry of 13 Poecilopora colonies. You can see the field measurement on the x-axis and the associated 3D surface area, in this case, on the y-axis. So we completed this process for 15 different morphotypes of corals, and we did that for both 1D to 2D relationships and also 1D to 3D surface area relationships, which involved the photogrammetry of 160 colonies of coral. And we're hoping to publish a paper on this work because we see a whole load of applications of these morphotype-specific conversions, including for things like carbonate budget modeling and things like that. So back to our feeding study. For all of the thousands of measurements of feeding scars that we collected from our surveys, we were now able to use our conversion metrics to convert each of these individual scars to their associated planar area and 3D surface area. So from this, we were able to work out mean daily feeding rates per individual by summing together the feeding data for each individual starfish and then dividing it by the aging factor, which I was speaking about before, which was 6 days for the summer months and 10 days for the winter. So finally, we had our mean daily feeding rates for each individual starfish. So for those of you in the room that work with ecosystem monitoring, modeling, or management, you'll probably be most interested in our data on 2D planar area of feeding rate, because these feeding rates can be related to total coral cover on the reef and will demonstrate the collected damage potential of two ecosystems if COTS are left unmanaged. And for those of you in the room that are perhaps more interested in nutrition and physiology, our data on 3D surface area consumption, which basically signifies total tissue consumption, will be of interest because this data tells us a lot about the underlying explanations for feeding differences between corals and also how these feeding differences may be affecting the physiology of the individuals. So let's have a look at some of our results. So in total, we were able to collect mean daily feeding rates for 540 different individuals, which involved the measurements of over 2,500 feeding scars. So this is by far the biggest COTS feeding study that's been done. We found a lot of variability within our daily feeding rate data set, and so we wanted to try and dig into what was driving that variation. So the most obvious to investigate first was the size of the effect of body size on daily feeding rate, and as expected, we found a significant positive effect of body diameter on daily feeding rate. And with that in mind, we were able to group our COTS into size categories and then calculate mean daily feeding rates for different size categories of COTS. And for ease of use, we've used the same size categories as what is used by the culling teams when they collect their size structure data. So I'll just talk you through what some of these daily feeding rates look like, because sometimes it's hard to sort of visualize that sort of thing. So for COTS in our smallest size category, these individuals were eating around 13 square centimeters on average a day, which is roughly the same size as a small biscuit. Then jumping to COTS in the 25 to 40 centimeter class, these COTS were eating on average roughly around the same size as a CD. And then COTS in our largest size category were eating 241 square centimeters a day, which is roughly equivalent to a frisbee or a small dinner plate. And that's each day. So if we think about that at a reef scale, the collective potential of these large individuals, which we know are making up the majority of our... I'll go back, actually. So we know that a lot of the individuals on our high-density outbreaking reefs at the moment are in the larger size categories, sort of bigger than 40 centimeters. So if we think about the potential for consumption, these large individuals are eating around 6 to 8 square meters of coral per year each. And if we multiply that by the thousands, tens of thousands of COTS that we have on our reefs at the moment, the coral loss potential from these populations is really phenomenal. So now we're just going to have a look at some of the other drivers of feeding rate, and for the figures that will follow, body size has been accounted for to affect that because of the confounding effect. So I'll be presenting relative daily feeding rate. So looking at the role of food availability on daily feeding rate, we found that total coral cover and aquapore cover were having a weak positive effect on relative feeding rate when total 3D surface area of coral consumed was plotted. However, interestingly, when we considered daily feeding rate to be the number of colonies consumed per day, as opposed to the area consumed per day, there was a negligible or even slightly negative effect of coral cover on daily feeding rate. So you can see those two graphs next to each other. So this is telling us that COTS in low coral cover sites are eating the same number of colonies as COTS in high coral cover sites, however receiving far less surface area and so tissue area per colony that they consume. This is likely because the sorts of corals that dominate high coral cover communities are taxa like Aquapora, which are highly complex, and so every colony consumed is offering a large amount of surface area per colony, whereas in low coral cover sites, the dominant taxa are often massive and submassive species, which are morphologically a lot less complex, meaning for the same number of colonies, the actual tissue consumption and nutrient intake would be much lower. We can also infer that for cots in these two communities to be reaching the same number of colonies, cots at low coral cover sites are likely foraging a lot further and exerting a lot more effort to receive food. So both of these factors are likely affecting the overall energetic condition of cots, which will undoubtedly be having an effect on the physiological condition of these populations. So now moving on to the effective region, we did find an effective region. However, this is most likely reflecting the fact that corals around the lizard region are dominated by highly complex corals, which is what I was just talking about. And we didn't find much evidence for an effect of population density or outbreak status on daily feeding rate, at least over the range of densities that we're currently experiencing. So what are the main findings and implications of this study? Our study used a highly accurate photogrammetry method paired with a huge sample size to produce accurate daily feeding rates for over 500 cots. With this data, we were able to show that cots' feeding rate is influenced by body size and to a lesser degree coral cover. However, population density was not having an effect on feeding rate. This study established mean daily feeding rates for several different size classes, and when these size-specific feeding rates are paired with our data on size structure and population density of known populations, this will enable massively improved estimations of cots' feeding impact for ecological modelling, and in particular will be a key component for refining cots control thresholds. Additionally, this study has recorded fine-scale data on total tissue consumption by cots, including taxa-specific consumption rates. This data will be used to better understand the basis of feeding preferences and, moreover, gain an insight into the physiological condition of cots during the course of an outbreak, which may ultimately explain population breakdown. Our improved understanding and management of this problematic species will provide the best chance for safeguarding the resilience of the reef. Thank you for listening, and thank you for CSIP for funding this research.
Thank you, Josie. That was a great talk. I've got two related questions. Firstly, if I remember that right, Morgan and others had found winter-summer differences quite big in the feeding. And secondly, I'm just curious, are your rates roughly in the same ballpark as the older rates from John Keesing? So we can still say maybe 10 to 15 per hectare is ecologically sustainable?
Yeah, so in response to your first question, yeah, we did also find seasonal differences in feeding rates, but these are most closely tied to the actual spawning, like localized around the spawning time of the year rather than the seasons like just summer and winter. So we found that we had much higher feeding rates in the month of spawning, and that wasn't exactly tied to temperature, for example. So yeah, I thought about including it, but didn't manage to make it into the presentation. And then with relation to Keesing's rates, ours at the moment are a little bit smaller than Keesing's rates. I think they're about 2/3, but we are in the range of what he said was normal. But he had a very small scale study of not very many individuals. So we do, as you can see, we have a whole range, and now we can see that we can pull it out given the size of the individuals and things like that. Anyone else?
All right, thank you very much. For our next presenter, we have Peter Dahl.
Thank you. I didn't have a very inspirational story. I never wanted to be a marine scientist, but then wanted to be an astronomer or a medical scientist, but then I moved from the Bavarian mountains to tropical Australia, and now I'm still researching coral reefs as a postdoc, so it's probably a bit too late to do 180 and go back to other things, and might as well continue to contribute my time towards resolving some of the critical information gaps that have accumulated in this space. You've probably read it already now. I'm talking about a project that's also funded by the CCIP, which investigates predation rates on adult Crown-of-Thorns starfish relative to fisheries management zones on the GBR. Some of the current collaborators for this project include Scott Ling at UTAS, as well as Sterling Tebbett and Andy Hoy at JCU. We all know that reef health and resilience are inherently contingent on moderating some of the major disturbances, such as the ones shown here in these photos. While slightly overshadowed by 5 major mass bleaching events in the past 8 years, it's important not to forget about these other ones, like tropical cyclones and Crown-of-Thorns starfish, which are the other 2 major contributors to reef degradation, both in the GBR Marine Park as well as the broader Indo-Pacific region. The problem is 2 of these 3 major disturbances or contributors to reef degradation, bleaching and cyclones, are disturbance events that we cannot really resolve or even effectively moderate at the scales most of us here operate, which leaves us with only one of these major contributors that is at least somewhat amenable to localized management efforts. Because Crown-of-Thorns starfish continue to devastate reefs and entire regions throughout the Indo-Pacific range, it appears critical to capitalize on the opportunity to build reef resilience by understanding their population eruptions and outbreaks and contribute to the ecological underpinning of COTS management. On the GBR, we're witnessing yet another outbreak wave. However, we still don't really know to what extent different natural and anthropogenic factors are responsible for these outbreaks. There are 3 hypotheses that have been around for a long time that have the greatest traction still in explaining these outbreaks. On the one hand, the natural life history of COTS, especially their extraordinary fecundity, predisposes them to undergo these major population fluctuations. And on the other hand, anthropogenic factors such as terrestrial runoff and the removal of predators have been proposed to likely contribute to the incidence and severity of outbreaks, at least in some locations. Let's briefly focus on the predator removal hypothesis, which is relevant for this current research that I'm presenting. The underlying mechanism proposed here is that predatory fishes or other predators that are more abundant within no-take areas because of limited fishing pressure are either directly or indirectly regulating COTS abundance and limiting the incidence or severity of their population outbreaks. This hypothesis was first posed by Robert Endian in the 1960s when it was focusing on giant triton, and he noticed that some of the first outbreaks of Crown-of-Thorns starfish were happening in areas where there was a lot of removal of giant triton snails, but it has since focused more or less on predatory fishes. In 2008, the seminal paper by Hugh Sweetman on the right came out, which showed that the incidence of outbreaks, specifically on mid-shelf reefs, which are those black bars, was proportionally much lower in no-take areas or green zones than in zones open to fishing. More recently, further analysis corroborated that outbreaks were less likely on reefs close to fishing, and this paper on the left by Frederick Kroon et al. Also reported that the abundance of predatory fishes was a lot higher in those green zones. And the paper on the right provided yet another line of evidence showing that the prevalence of sublethal injuries was higher in green zones. So there appears to be somewhat of a zoning effect, at least based on spatial outbreak patterns. The problem is that we're still lacking some of these critical links in our understanding of these patterns, as we're yet to quantify or compare relative rates of predation or risk of predation between areas closed and open to fishing. So the current study intended to fill this gap and compare risk of predation within different fisheries management zones. And we obviously also wanted to relate any differences in predation to the relative abundance of potential predators. The 2 study regions were the Lizard Island region in the northern GBR and the Townsville region in the central GBR, each including reefs within green zones close to fishing yellow zones where fishing is restricted, mostly to recreational fishing, and blue zones which are essentially open to fishing. We recently wrapped up the field data collection for this project, and over the past 2 years we've conducted 78 predation assays across these 6 locations, which include observations for more than 312 starfish. So that's the basic setup. So we set starfish out near the edge of the reef, and then we basically go back 24 hours later. That's sort of the time frame we worked out, and go back and score predation. So that includes partial predation. So if a starfish, for example, lost 3 arms, it would get a score something like 0.2 or 0.25, depending on the body size. And then a total predation, so the entire starfish was consumed, would be a score of 1. We also set up time-lapse cameras to try and capture predation events and identify the key predators. At each of these sites, we also conduct comprehensive surveys of the local fish and benthic assemblages, which of course include the abundance and size of all potential predatory fishes. And we also estimate the local densities of adult cots in the area using the salad method, because the role of predators will likely vary depending on the local densities of cots. When deploying the starfish, we also record a range of other factors, including the size, the reproductive status, sex, if applicable, and any pre-existing injuries. Of course, this kind of predation assay does not provide us with absolute rates of predation or mortality that we can just take and plug straight into population models. But it does give us unprecedented insights into relative risk of predation, as well as the identity and abundance of key predators. So that's probably a good point to circle back to this morning and thank Guju Guju for raising the important questions: who are the enemies of crown-of-thorns starfish, which he highlighted in his welcome as the hot topic in reef resilience work. I did not ask him to say that, but obviously it very much highlights the importance of this question and this research that we've been conducting. So let's have a look at some of the predators we recorded using the time-lapse cameras. Of the total predation recorded, 55.3% was ascribed to just the Spangled Emperor, Lithrinus nebulosus, and another 35.5% of predation occurred when both Lithrinus nebulosus, one or multiple, and a starry puffer, Acanthaster stellatus, were captured feeding. Together, these 2 species contributed to more than 90% of the predation recorded across all those predation assays. And up the top in red, you can also see that another 6.1% of predation was ascribed to the combination of Lithrinus nebulosus and the Titan triggerfish, with only 3% remaining for all other putative predators of adult starfish, which included another pufferfish species and 1 giant triton. What sticks out when looking at this pie, as well as the actual underlying footage, is that it appears that Lithrinus nebulosus is in pretty much all cases necessary to get the predation party started, and then in many cases the starry puffer sort of joins in at the same time or shortly after. These 2 species were the only species recorded to consume entire adult starfish, which included even some of the biggest individuals that we deployed. Aside from these, we've recorded various other species taking bites, but only after there was already significant damage to the individual, especially when things like Paloxica or gonas are floating around. That's when butterfly fish and everything comes in. But it does suggest that out of the many dozens of fish species that have been in the past proposed to feed on adult crown-of-thorns starfish, most of them are just scavengers and do not actually kill and consume entire adults. Across all sites and zones, the mean predation rates recorded to date was about nearly 20%, ranging from 0% where none of the starfish were touched at all to 75% where 3 out of the 4 deployed ones in that predation assay were fully consumed. Forgive the very basic figures at this early stage, but I think the data speaks for itself. Importantly, when comparing the overall rates of predation among those different fisheries management zones, we can see that predation was 3.6 times higher in green zones compared to blue zones and 2.8 times higher than in the yellow zones. Clearly, the adult cots deployed in the no-take areas were at substantially higher risk of predation, supporting the zoning effect and the predator removal hypothesis. Now looking at the zoning results, but split by region, clearly the zoning pattern we detected is quite consistent for both regions. For the Lizard Island region, the northern GBR predation was 3.5 and 3 times higher in the green zone compared to the blue and yellow respectively. And for the Townsville region, the central GBR predation was 4.5 and 3 times higher compared to blue and yellow respectively. So the logical next step is to analyze the fish community data and other factors such as the size and the reproductive status of the animals. And other important link to test will be to see whether these clear differences that we found in predation relate to the spatial variation in the abundance of key predators, particularly based on these results, the spangled emperors. Some early key takeaways from this work so far include that it shows that we previously vastly overestimated the number of predatory fishes that can kill and consume adult starfish. It appears to be really just one primary predator and a couple more in total, at least at the reefs and sites and regions we worked at, with other fishes previously believed to consume cods likely just being scavengers at best. The overlap between predators and fisheries target species is also quite low. Common target species such as coral trout were never observed to feed on any of the starfish. At the same time, the starry puffers and the Titan triggerfish are obviously not fisheries target species. The only overlap really seems to be the spangled emperors, which are a popular target of the recreational fishery and as well as a secondary species for the reef line fishery, which could be part of the reason why our study demonstrated that cots experience a substantially higher risk of predation in no-take areas than in blue and yellow zones, so that's where potentially that recreational part comes in. Our results corroborate the hypothesis that no-take areas and limited fishing pressure could regulate cots abundance, at least in some locations. These data on key predators and zoning effect also provide opportunities to improve the ecological underpinning of both modelling as well as management. Ecological data corroborating the zoning effect of outbreaks could be useful to model population dynamics and refine ecosystem models for targeted decision support. Lastly, these data also allow us to assess the potential role of fisheries management and zoning in moderating COTS outbreaks, which includes potentially the increased abundance of the predators identified in this project and other projects identified in the next talk, particularly for the adult stage, the spangled emperor. Thanks for the COTS Control Innovation Program, as well as the Lizard Island Research Station and all the crew, collaborators and researchers involved in this work. Thanks.
I'll just check in online. All right, we'll come back to that if we have any online questions.
A great talk. If we flip the question a bit to the predator as opposed to the prey, if fish emperors are really kind of the only ones that eat the adults, that would suggest they could benefit a lot from outbreaks, right? So if they're monitoring data on their abundances, and do they go bone and bust with the outbreaks of COTS?
Yeah, that's a great question. I guess there's two sides to this. Obviously, spangled emperors, similar with starry puffers, have a pretty general broad diet, so they're not just specialised on Crown-of-Thorns starfish and feed on various inverts. But at the same time, when we look at monitoring data, there isn't really much on these species. I'm pretty sure, correct me if I'm wrong, but the LTMP doesn't record spangled emperor and starry puffers and some of those predators, so we'd probably have to look at fisheries data, potentially, which obviously has some limitations to it in interpreting and matching up these fluctuations of both COTS and the fishes. So yeah, and our own sort of site-specific fish surveys are obviously too short-term to dig into any sort of temporal variations in the fish abundance, unfortunately, but it's a good point.
Sorry, I might just build on that one. So my master's degree in the early 90s, like 93, we looked at fishing club records from the 50s to 1990, and there was trends in some of the target species, like Lathrinids over, I think it was 7 or 8 year periods, so it was quite cyclic. But my question is, so there was Townsville and Lizard Island. There's a huge amount of difference in the fishing effort, particularly recreational fishing, between Townsville and Lizard Island. I'm just wondering if you tried to standardise the actual fishing to tease out, is it a zoning effect or is it just the location where the zoning was applied, and look at some of the historical differences. And it might be a case that there's, for a small number of sites, it might be just it was just one of those things. We put the zones where it was, and there was a lot of negotiations around where the zones went, and a lot of background to that. So, yeah.
Yeah, no, I think halfway through the project, there seemed to be a bit of a difference in the yellow zone. But now that we've done plenty of trips and a fair bit of replication, it's sort of balanced out, and we see that same green, yellow, blue effect at lizard as well. In the first place, we put it down to obviously a lot more recreational fishing at places like John Brewer, I think is the yellow zone, as opposed to up north. But yeah, in general, it would obviously be good to do this entire thing across the length of the GBR and have like various data points. The fact that we only have 2 probably rules out too much emphasis on that, but if we had a bit of a gradient, then there would be a lot more we could do in terms of standardising data.
Final question. Do you just put a 360-degree camera on a stick and glue it to a starfish, or what was going on in those photos?
No, so these are sort of mounted cameras, and we put them on time-lapse mode. I think AIMS developed some really fancy things that we trialled once at Lizard Island, where you got 24-hour continuous recordings of like a 2x2 metre area, but unfortunately we didn't have that available for most of this project. No, these are really basic time-lapse cameras, just focused on the deployed starfish to try to capture as many events and predators as possible. But yes, of course, it would be ideal to have a very realistic, massive arena, and we could sort of just let them run around. But yeah, with what we had to work with a few years ago, we thought this was the most appropriate way to try to catch as much action as possible. Great, thank you. Thanks.
For our last talk, we're welcoming back Pete Mumby in a different capacity, switching to the COTS side of things, and then we're off to lunch.
Yeah, good morning, everybody. I'd like to present this on behalf of the team. So this work was led by postdoc Kenny Wolfe and PhD student Amy Despiens, with a bunch of great collaborators, including Eric Fisher and Sven's team. Okay. So we're going to talk about this juvenile phase of COTS, and those people that aren't familiar with it will learn a lot more at Maria's talk later, I'm sure. But as many of you know, Crown-of-Thorns starfish juveniles often recruit into rubble, where they are mostly herbivorous, feeding on CCA, until they get large enough to become corallivores and start eating coral. But of course, we know relatively little about what happens to them during this period in the rubble. And rubble is something that we've been studying for a number of years now. And so the first question we had was, well, what of the things that live in rubble might actually eat these starfish? And this is the first question. It's part of Amy's PhD. It's very simple. You just take lots and lots of creatures you find in the rubble and ask, are they able to consume starfish? And the outcome was, after testing over 100 taxa in 500 trials, there was nearly 30 species that had some ability to consume crown-of-thorns starfish, but it varied a lot among species. And really that there's 2 major groups. There's one primary species, and this is Schizophris aspera, the decorator crab, and this is the sort of Rolls-Royce of starfish consuming invertebrates in the rubble. And then at the same time, there are the swimming crabs, the portunids, which you'll be familiar with. And they have a much lesser role, but they're still important. So the first question then is, yeah, these 2 groups are pretty significant in terms of their ability to consume. The next question is, yeah, but do they even bother to consume crown-of-thorns starfish? And to do that, we had some mesocosms. So we just collected rubble from the reef, put them in these tanks, and that provides lots of hiding places for these tiny starfish. And they're really pretty hard to see and to find in these environments. When you add the decorator crab, they're able to consume 5 starfish per day. So they're able to find them and consume them quite readily. And if you look at the proportion of the starfish that are being consumed, it's pretty high. Up to 80% were being removed by this single predator species in these mesocosms. So despite all the vast diversity that lives in rubble, there's this one crab that does the lion's share of it, and another group that have quite a significant effect. And, it's not a simple process. If you look at the probability that a baby starfish is completely consumed by a crab, it declines as the starfish gets larger. That's not a big surprise. And generally, you find that once the starfish is about 1 centimeter in diameter, it's no longer being consumed absolutely by the decorator crab, even less so by the portunids. But what happens instead is that the starfish get damaged. And as they get to this sort of size of 1 centimeter and beyond, you're seeing more and more damage, arms being cut off, physical damage to the starfish, particularly by aspera and to less so in the portunids, although that is their main mode of action. They cause damage to these starfish. And interestingly, that sort of aligns with the work of Keesing, which found that mortality rates of starfish drop quite significantly once they reach a size of 10 millimeters. So that seems consistent. So the most obvious question that follows from this is, well, all right, if you've got natural populations of these starfish in the rubble, does that in any way reduce the likelihood of a starfish outbreak? Or maybe even not an outbreak, but just reduce the population size of the adult starfish? And this is an incredibly difficult thing to measure empirically. So these data are just indicative. But this is data from around Heron. And so Kenny and Amy have gone around Heron Island here doing extensive surveys for the abundance of these crabs. And when you look at the relationship between the density of crabs and whether or not there were large populations of adult Crown-of-Thorns starfish, as found in the COTS control program, you find this negative relationship, which is what you would expect if areas that seem to have few predators tended to have the most adult starfish. So it's sort of indicative that there could be this control happening to some extent, at least around Heron Reef. And then working with Sven's team, we can get into the mechanics of it. You can ask, to what extent can you even detect that these crabs are consuming starfish readily? And so using eDNA, so by providing the crabs starfish prey, what we find is that there's 90% detection of COTS DNA in these crabs within 12 hours of them eating it. So as long as they've had a meal within the last 12 hours, you'll know about it if you happen to sample that crab. So that's good to know. And interestingly, of the 13 wild-caught schizophrits found around Heron Island, 2 of those actually had COTS detected in their stomachs. So they are actively feeding on juvenile crown-of-thorns in the rubble at the time that we were doing those surveys. But how do you generalize this? And this is where the story becomes intriguing. This is what you see in terms of the abundance of these crabs at Heron Island. And so typically, there's a reasonable abundance of schizophrits, and there's a greater abundance of the portunids, even though they do less. This is Heron Island story. Some relatively brief surveys with Eric's team at Moore Reef showed a very different picture. And then extensive surveys at Lizard reinforce this picture, that there are far fewer crabs being found at Lizard than we're seeing around Heron. And that raises some very interesting questions. So we know then that it seems schizophrits are relatively uncommon in these areas that have been surveyed in the northern GBR. Is this a general trend for the northern GBR? We don't know, but it could be. It doesn't seem to be a biogeographical biodiversity story because it's the same species involved in both Heron Island and at Lizard Island. It's not like we're serving a different community. It's just a more depauperate community of predators. But one of the things I was chatting with Amy about this, her feeling was that, and she has seen an awful lot of rubble beds over the last few years, and her feeling was that the rubble beds are different. She told me that where they were finding aspera, it was in the rubble beds that are like the rubble beds around Heron Reef. And those are the consolidated, quite well-structured rubble that's been there for some time. Typically, that rubble doesn't have much sediment associated with it. At Lizard, there was lots of very crumbled rubble, lots of sediment associated with it. It might be that that is a very bad rubble habitat for crabs. We don't know necessarily if it's a bad rubble habitat for starfish. So is the problem the type of rubble? And how widespread is this issue in the north? To what extent do these different types of rubble beds impact starfish as well as their predators? We don't know. So I think what this tells us is that there's clearly a link between predation and COTS populations at that small scale within the rubble beds in the south. There's indicative evidence that this might have demographic consequences in terms of the regular COTS adult abundance. But as you start looking further north and more widespread, the picture becomes pretty unclear. And it means we really need to investigate what's happening in this rubble much more extensively now, because there is the potential for an important link that connects very nicely with the work that Peter just presented on the fish predators. And just like to thank our key collaborators, in particularly the Gadajul Development Corporation, with whom we've been working in around Heron Island. Thanks a lot.
For those of you who are avid rubble type definition fans, come back to this room tomorrow. I'm not biased. There's a question.
With the nibbling of cots at a very small size, do they show autotomy at that point? You know, do they slough off legs, or does that happen later in life?
What Amy is seeing, so she's looking at the survival of these starfish once they're being sort of having these interactions with portunids and things, and she is seeing significant mortality in some of those corals once they're damaged. So it's not so much that they're sloughing off legs, but that process is an important source of damage and affects their survival. Yeah.
And just with the rubble, the mixed clastics with more sand versus the rubble, do you get more COTS juveniles in the rubble versus the mixed clastics? Or?
Yeah, that's right. So, yeah, when you see a sort of beautifully clean rubble patch that's quite well developed, usually quite thick, that's the sort of rubble that you will find these predators under. They'll be typically underneath a slab of table coral, something like that. Yeah, but that's what they like. There's presumably enough flow in that system for them.
And we have an online question. How long does it take the COTS to reach 1 centimeter? What kind of time window do the crabs have for predation in the wild?
Now that is a question I have to defer to come back and listen to Maria's talk, because it's not a simple answer. Right? We don't.
About 4 to 6 months, depends on the COT.
Okay.
4 to 6 months.
Yeah.
Yep.
It depends whether they have coral available to switch off or not. So yes, for me as well, it's roughly 4 months.
We have another comment that the diet and coral availability affect that window.
Pete, this is super interesting. I'm thinking about Jennifer Wilm's work on the rubble, and she was working at 10 meters or something, or 5 meters. Anyway, the rubble that she was working on, which is crustose coral and algal-covered rubble, is probably quite similar to the rubble that Kenny saw at Heron. So maybe where they looked at the rubble on Lizard and Moor Reef is probably not where where the juveniles' COTS are in Moor Reef, but would the rubble that Jennifer Wilma saw be more similar to the rubble where Kenny saw crabs on Heron?
Quite feasibly, and I think this is where we need to get everybody sitting around looking at pictures of rubble and agree what sort of rubble is rubble A and what's rubble B. And, this is one of these problems that there's a whole bunch of people who've been working on rubble, and rubble means different things to different people. We don't have a good classification of it yet, because it's more than just the shape and whether it's colonized by CCA or not. I mean, that's going to influence, obviously, the suitability for food source. But whether or not that can support the structure that will allow a high diversity of predators is another story. So, I think that there's a lot to be gained by bringing people together to figure out just how common are these rubble habitats. Because we see huge differences even within heron. You can go to an area that has more sediment and it functions completely differently. So it's pretty interesting.
I think we'll wrap it up on that note and head off to lunch. Thank you very much to the speakers this morning.
Island and Cairns, and also much further north in Princess Charlotte Bay, and the reefs we nominally refer to as Cape Grenville. So that little spit sticking out just below the red box is Cape Grenville. So these are actually right up in the far north of the Great Barrier Reef. So the project was very much focused on providing critical information about when or where the fifth documented population eruption might arise, and the intention was to maybe facilitate early detection and perhaps even preemptive culling to stop this outbreak before it got started. In terms of the method, you've probably heard a bit about this already if you've heard some of the other COTS talks, but we're using large-scale surveys which are undertaken on underwater scooters. So traditional methods, whether it's the fine-scale transect-based surveys or the manta-tow surveys, are very good at detecting crown-of-thorns when there's outbreaks. The problem is both these methods, for different reasons, really struggle in the detection of low densities of crown-of-thorns starfish. So we've used a sort of mixed model approach where the diver has the full autonomy to stop, check whether feeding scars are actually caused by crown-of-thorns and also search for the crown-of-thorns, but also has the capacity to travel up to 2 kilometres in a single dive to cover very large areas. So using this method, we've been able to track relatively low density populations of crown-of-thorns. And so the best demonstration of this is the change over time that's apparent at Lizard Island, noting this is where we first established this technique and we first started sampling in the latter part of 2019. So we have a fairly continuous record. So you can see the vertical lines represent the timing at which these were conducted. There's definitely variation in the recorded densities around the toes, but the general pattern is that there were relatively moderate densities, around 8 starfish per hectare up until the end of sort of 2021, which may represent the baseline sort of densities of crown-of-thorns you expect in this area. Then from that point on, we've seen this sustained increase in densities to the point where in the latest survey, the mean density is very much approaching the traditional threshold for what we consider population outbreaks to be at around 15 starfish per hectare. Unfortunately, this is the end of this project, so this was an unprecedented opportunity to really track the progression of these outbreaks, but at this stage, there's no plans to continue this work. Now, looking further afield, the key thing was that while we were seeing that build up at Lizard Island, the elevated densities were already apparent in the far northern part of that range. So the red box there up at Cape Grenville, when we did the surveys, expanded the surveys to all those 4 regions, we were already seeing elevated densities up around Cape Grenville, whereas the densities at all other regions were down around that 8 or less per hectare. So it seems that, if there was an initiation, it's most likely that it probably started well north of Lizard Island. So perhaps we need to rethink the area of the putative initiation box. And to add further evidence to that, when we did this initial sampling, the size of the starfish was much larger up at Cape Grenville. So if you just look at those modes there, the mode of the starfish for Cape Grenville was over 40 centimetres diameter, whereas at most other locations they're much smaller. So it does seem that it is most likely that we had elevated densities of large starfish up in the far north, and they may have been reproducing and sending their larvae down towards Lizard Island. So it may be that Lizard Island, which has been our focus for a long time, is actually just the receiving area for larvae from established outbreaks in the far north. So in terms of the key findings, thank you. So we're seeing renewed outbreaks of population eruptions of crown-of-thorns representing the 5th documented episode on the Great Barrier Reef. These are already apparent. So while we're now seeing, a lot of coral mortality due to things like the recent bleaching, we're also seeing the initiation of the 5th major outbreak of crown-of-thorns starfish. These recurrent salad surveys have definitely provided some unprecedented insights into non-outbreak or pre-outbreak populations, suggesting that the baseline densities may be as high as 8 starfish per hectare, which is what we saw at a fairly constant level from 2019 through to 2021. Now, there's been sustained increase since that time, which is the evidence to suggest that we are well on our way to seeing the establishment of the 5th major outbreak. Now, this sustained increase in densities at Lizard Island has occurred despite ongoing culling in this area. So elevated densities of Crown-of-Thorns starfish were already apparent on the reefs to the north of Lizard Island, suggesting that, it may actually be that the putative initiation box is either much larger or centred to the north of where we've previously focused a lot of our attention. So in terms of the take-home messages, these novel sampling methods, and particularly the scooter-assisted surveys, enable much higher detection during large-scale surveys, providing unprecedented insights, especially during the build-up or pre-outbreak situations. And this is important because it's provided probably new insights where previously we just detected Crown-of-Thorns outbreaks once they were very well established in any given area, where all of those techniques are very valid for saying whether there's an outbreak. It's the subtle changes in density and size structure that happen in the build-up that we can't really detect otherwise. So it gives us new focus to really think about the northern section. And finally, it does point to the fact that the established culling and surveillance methods are probably ineffective for really understanding what's happening in those low-density situations, and greatly constrain our capacity to really contain these renewed outbreaks moving forward. Thanks very much.
Thanks very much, Morgan. I can see a question from Leah.
Yeah. I intentionally haven't talked too much about that because that very much is Sev's domain in this particular combination of projects. The sort of take-home message there is that at least half the time the currents do flow north to south, even in that region, whereas for a long time the prevailing paradigm was that you had that Vanuatu jet which hit the GBR around Lizard Island and then sent a current north. But there's also the opposite that we are seeing happening. So as part of CSIP, we've deployed current meters through that entire region whenever we've been up there.
Mariana?
Morgan, the Cape Grenville's cots were very, very large, so they're very, very fecund. Now, what I didn't quite get it, they weren't in a big aggregation. They were just very large and low density. Is that what you're saying?
They're much higher densities than at Lizard Island at the time. And the densities are around that 15 starfish per hectare. The interesting thing is I expected them to continue to grow, but they haven't really changed that much over the entire period we've been going there. They haven't crashed, they're still there, and we actually thought they were running out of food until this latest survey where, the wind was from the north, and so we were diving on the south and found a massive area of very healthy staghorn acropora, and not surprisingly, there's cots all through there. So it's an ongoing problem up there.
Because I was thinking that if they're very large and fecund, you wouldn't even need many of them.
Absolutely. Yeah.
You don't need an outbreak.
That's right. Yeah. So our latest estimates of fecundity are close to 1 billion per starfish. So you need 1 male and 1 female to get the entire population that constitutes an outbreak across the entire GBR.
Thanks very much, Morgan. Our next speaker is Sven Uthicke from Ames. Thanks.
Thanks, Katharina. I will also briefly share some of my personal history because it's the topic of the day. As Peter and Katharina, who both presented this morning, I was also born in Germany. I was fortunate enough to be born a little bit closer to the sea than these two. I'm from Hamburg. I initially got into diving and marine conservation because I think, like many young people of my generation, adored Jacques Cousteau, and so I watched Jacques Cousteau movies, so I must admit I was initially a Mediterranean fan, and I only coincidentally came to Australia, to Lizard Island as a volunteer, and fell in love with the Barrier Reef, and hence I'm still here working at AIMS. So I'm going to talk about a CSIP project we're running at AIMS, but also a lot of in collaboration with Morgan's team, so some of the topics I'm going to talk about may sound a bit repetitive to you. So we're using eDNA as a new monitoring tool with the same aim Morgan has to detect really early stages of outbreaks. So as I mentioned, it's a team of us at AIMS in collaboration with Morgan's team and also with Emma and people at CSIRO. And it's CSIP project C03. As Morgan alluded to, it is assumed that the outbreaks start in this outbreak box, but we know now that it's possible that they start much further north. They slowly move down south, as they are now basically in the south of Townsville, and we have an ongoing permanent outbreak in the Swain regions. However, there's now indications that these are further north. As also all of you know, there's an active culling activity, huge culling activity at the GBR run by Gabrumpa and the operators. And the main idea of CSIP is to support these culling and optimize the culling activities and the management of the cots on the GBR. I don't have to spend much time on explaining manta toes again. It is, as Morgan said, a good method to document existing outbreaks, but it's not fine enough to find early numbers. So in addition, low densities. In addition to the salad surveys, we had started at AIMS to develop eDNA techniques to monitor cots. eDNA is a technique just in general where you would catch free DNA in the water, which could still be associated to sails or could be freely in the water or absorbed to other surfaces, and you use various different techniques to identify those. In terrestrial and freshwater environments, this has been used quite frequently since the last 15 years for general biodiversity descriptions or detection of introduced species, but also in quite exciting studies on documenting the ecology of ancient species which are extinct, like the mammoth or the moa in New Zealand. There's a few different techniques which is usually confusing people a lot who are not in this field. One of them is metabarcoding, which looks at multiple species and usually uses next generation sequencing. This is a technique which gives you many species, but it's probably not, or only to a very limited amount quantitative. However, what we do is single species detection with specific markers designed to only detect that species. And that can, at least to some extent, be quantitative. There's two methods, which is one is quantitative PCR, the other one we're using digital PCR. So you can quantify the number of copies in the water, and you can later on relate that to actual densities in the field, if you're lucky. We have, as I said, worked on this for over 10 years. We have already used this quite extensively. We have an ongoing larvae monitoring program, which is supported by lots of the culling people represented here. So most of the COTS control boats are collecting water and plankton samples for us so that we can monitor the presence and abundance of larvae. And we've used this for juveniles and adults. That's what we're going to talk about here. Peter Doll, as part of his PhD, has used our eDNA technique to quantify newly settled juveniles on the reef. And as Pete Mumby related, we are also using this to look for predators in the gut contents of these predators. We're looking for identifying crown of thorns. But here, as part of CSIP 03. We're going to focus on adult and juvenile detection. Briefly, the workflow is you have to go out in the field. We're collecting 3 sites on every reef at the moment, taking 12 samples on these filters. So 2 liters of water are filtered over the filters. They're fixed in a fixative, and in the lab they're extracted and the DNA quantified. We had published 2 papers before we started the CSIP work. One was at really high COTS densities, where we showed a rough correlation between eDNA and COTS. The second one, just when this project started, we had looked at lower density populations, including some of the salad data at that stage, and we showed that the proportion of positive samples is quite well related to the actual density of crown-of-thorns, as depicted here. So the main purpose of this project, so the method basically existed. The main purpose was to test a large-scale monitoring, which we implemented on 16 reefs over the last 3 years. This monitoring was complemented by samples from up north. Morgan's team has collected as part of a cooperation with PO4, and we also later on added samples through ships of opportunity, specifically testing whether, for example, the Queensland Parks and Wildlife boats could collect for us and add to the data set. Jumping straight into the results, so the results can be shown like this. This is the first year of surveys we have conducted. You see roughly 20 reefs presented. The more red you have in those little pie charts, the higher proportion of positive samples you have. Now you might wonder, and I'm going to explain that later on, what does that mean? The more red, the worse, right? Any completely white pie chart has no COT eDNA detected, whereas if it's completely red, all of them the whole pie chart's red. So you see in the first year, even up in Princess Charlotte Bay in Cape Granville area, we only had 1 reef which really had high densities, and which is the completely red reef there. And down at the south, you see the ongoing outbreak still documented at Davies and Bowden Reef, and high densities of COTS there. In the second year and third year, the number of red dots had quite increased, so the second and third year really high densities comparable to Morgan's data in Princess Charlotte Bay and in Cape Granville sector. Even the last year looked like it's getting a little bit less. You see how in the last year we really increased our sample size, which was not through increasing our own effort, but by adding samples other people like Queensland Parks and Wildlife have collected to us. And we also had a collaboration with Emma's group on calibrating these 2 methods, so that set another set of data there. So I'm hoping this shows with relatively small effort you can investigate and test a huge number of reefs for crown-of-thorns in relatively fast time. Yes, the analysis does take us a bit in the field. Also, thanks to the collaboration with Morgan, we have now over 40 reefs where we can compare numbers we are taking with the numbers his team is counting in the field. We can derive a relatively good correlation between, or regression between, crown-of-thorns densities and our eDNA results, which means we can put this into practice and decide, for example, if we want to detect COTS at the 3 number per hectare, where would be the threshold be? Just for argument's sake, we say maybe we should say if more than 30% of the 40% of the samples are positive, there's an outbreak, which would mean, but these numbers are debatable. We could say maybe we just want to detect 10, so we could set that up, or maybe we want to have a more conservative estimate, so we set the number down. But just for argument's sake, that would mean we could translate the pie charty bars and charts into really easy numbers, basically just in a binary matrix where we can convey this to management, that we think on these reefs, which are depicted red here, are the reefs where some sort of management action should happen. Is it culling, or is it sending out more survey teams doing detailed salad surveys, or some mix in between? So in the end, a complex method, but it allows fast detection, and we can work on a lot of reefs at the same time and analyze, but we can distill it down to quite simple messages needed for management. Also briefly mentioning Lizard Island, because that was one of the, what we call a sentinel sites, similar to Morgan's team. We have data since 2019 from Lizard Island, and you see at the top graph that roughly at the same time when the salad data jumped up, the eDNA concentrations also jump up. The top line is the average of the whole island, whereas the bottom line is 5 different locations. With exception of the lagoon, which always has really low numbers, you basically see an increase in DNA in all 5 sites, which is really quite well corresponding to Morgan's data and the ones Josie Chandler has talked about before and already published. Uptake of the project, I think we are really closely collaborating with Gebrumper and with CSIM and with the operators. We're annually reporting the data to Sam Matthew at Gebrumpers. They're all, the first 2 years are available in the COTS dashboard. They're used in a test case for an early detection system. So the uptake is quite well, and I think we conveyed well how these data can be used in the monitoring. Recommendations and conclusions. So similar to the salad surveys, we can clearly say the 5th outbreak has started. It is very likely that it started in Lizard Island, and it looks likely that it has started further north, as Morgan alluded to, which is really important to understand, because all the outbreak causes we are discussing, be it nutrients or predation, are really quite different if you're looking at an area further north. You could nearly argue both nutrients and predation should be less of a cause up there, but that's a case for further studies. Just making the point that we really need to know where these outbreaks are to start speculating or researching what's causing them. I hope I convinced you that the method is quite cost efficient and fast, and that with using ships of opportunity, we will make this even more efficient. It's by no way anywhere in competition with salad surveys. I think they're 2 very complementary methods. Our method could easily be used to measure several hundreds of reefs annually, basically, and then you could focus on the reefs where we recommend action with, for example, further salad surveys. The recommendations we will roughly spell out in our final report will be to continue this backbone monitoring on roughly 20 reefs, so that we can see how patterns shift over time. Also to have this monitoring complemented by ships of opportunity, such as the Joint Field Monitoring Program and other data. And we will likely also recommend to continue the Lizard Island data. As for Morgan's team, currently there's no funding to do that, so we're hoping to be able to do that a little bit over the next few years. And in addition to that, we propose to not stop the research and continue development. There's actually quite good uptake at the moment. I'm quite chuffed that even 2 publications from China are citing our method, and that these guys have developed what's called a biosensor based on COTS DNA. So basically just some probe you can stick in the water and get a yes or no answer. So that's obviously the next step, and we're hoping to take that on board and further develop our technique. Thanks.
Thanks very much, Sven. Are there any questions for Russell?
Okay, it's two separate questions, Russell. Firstly, yes, there's only there's two Acanthaster species in the GBR. One is brevispinus, and the other, the one with many names. It doesn't detect brevispinus, so it really only detects the coral-eating cods. However, it would detect the other cods from the Indo-Pacific area. To look at sea cucumbers or any other species, you would have to redevelop new primers and revalidate how they work, yes. But it is something we're proposing for a while, that it's used for a larger monitoring.
How much work is developing new primers?
Sentence with a piece of string comes to my mind, but I can't. It could be a year of a technician's time, yes. It's not, ideally it's been done for multiple species at the same time, but it is, with cross-test, it's more than a master's, put it that way. Or maybe it's a master's, roughly, year. Anybody else than Russell?
Rena?
Hi, Sven. Thank you. That was very interesting. I'm wondering what the spatial reach this method is. So if you take a sample with this very program lizard and you detect certain density, how can you know that's for a rock lizard or a nearby reef?
It's a good question, and it's something, thanks Renata, something we're looking at quite intensively because we're getting it asked a lot. In the moment, we're statistically analyzing how well our reef-wide data match to the salad data, which I think I showed matches pretty well. If you look at locations, that's getting a little bit wobbly. It's not as great. So I think it's a reef-wide estimate at the moment. However, we have done transects across the reef starting from a kilometer windward to a kilometer downwind. Practically windward, we're not detecting any. Then we're hitting the reef like Davies, you get a signal and you get half a mile behind the reef. You might still get a footprint, but it's pretty quickly diluting.
Okay, I think we're out of time.
Thanks.
Thank you, Sven. And our next speaker is Zeeshan Hayder from Seastar. Oh, thank you.
Thank you. Hello and good afternoon, everyone. First of all, a little bit introduction about myself. My name is Zeeshan Hayder, and I'm from Pakistan. I was born there. And around 10 years ago, I came to Australia to do my PhD. I'm not a marine biologist, or I don't have a background in biology, but I'm a machine learning guy here. I did my PhD in computer vision and machine learning. And the kind of the work we do here is like mostly object detection and segmentation. So we are working on different sort of applied AI things. So our AI methods can be used not just for like the, you can say underwater applications, but we are also using on the land and on the space as well. Because the similar methods are being developed and it can be applied like for across like different sort of applications here. And those are the things, new technologies that I want to discuss here. So that's why our topic is actually application of new technologies to coral reef monitoring and cost detection. So what are the problems that we are actually trying to solve here in this scenario? So when we look at this cost detection thing, the first challenge is that how can we solve this whole problem efficiently and accurately, and we can scale this thing onto like multiple sites and multiple locations, and without the need of any intervention from humans sort of thing. So that is our like first task, and how can we like train some AI models that can resolve, that can do all of this object detection automatically. Second thing is that all of these existing methods that were like developed in like last 40 years, they use some sort of control from humans, like for example, these Metato methods that are being used these days. They require some experienced and trained observers. So it is like too much reliant on human at the moment. And also, these methods cannot be used in areas which are like pest-infected areas due to some safety issues. So what are like more safe ways to monitor and document all these sort of course outbreaks? So there come all these new technologies that we can use. Okay, so in this scenario, what kind of technologies that we can provide that can help all of these court detection things? So first of all is that we have like 3 different parts to our work that we are doing. So first thing is that we are using a towed camera platform that we are developing with the help of AIMS. So AIMS is developing a platform that is like much faster and very safe, and it can only require like much less number of trained observers so that they can operate it themselves. Secondly, is that how we can develop some AI and machine learning technologies that can identify cuts and scars in the images. The main goal in this scenario is that we need some instant feedback for the observing team, and how we can reduce it and remove the need to manually analyze the images. So, for example, we have like a much bigger area, and if we can like capture the video and do all this analysis on the boat itself, on the vessel, in the real time, so those kind of technologies can be very helpful in this scenario. And thirdly, it can also provide some actionable information that can help the culling work and different monitoring teams work as well. And the third thing is that how we can use this cloud computing and data workflows in order to consolidate and deliver results in like much more consistent manner. For example, it will help to reduce the workload of the existing data collection and data backup methods. Also, how we can simplify these reporting things and ensure that everyone can be using the same sort of data that we are collecting. And also, if we can make these data public, it will be very useful for other computer vision or machine learning scientists so that they can use the same images and develop much better methods for the future. So here is like a simple real-time workflow that we are working on. So we have a vessel and an operator is actually controlling that thing, and we have a towed autonomous platform, which is like attached to the vessel via cable. So this cable is actually transmitting the data and control from that one to here. And actually, it is actually detecting cuts and the scars real time, and it is providing that feedback to the observer on the vessel. So as I discussed earlier, that we have 3 components in our system. So the first component is this new towed platform. So AIMS is actually developing this one. This platform consisted of like a small camera which is tied to the front of the platform, and it is actually doing 2 things. One is that it is providing images to the operator so that it can steer this vehicle and it can see some obstacles which are in front of these one, and he can like maneuver these things. Second, he can actually control the altitude and depth of the vessel, for example, in order to have like a better clear view of the scenarios. Third is that all the images or data that we are collecting from this platform is actually GPS tagged, so that we can exactly see that, okay, which location has like more cores and like scars in the images. So on this platform, we have like a small AI computer, which is actually doing analysis on board. So it doesn't need to transmit all the images or videos back to the cloud or do some offline processing. No, it can do all of those sort of object and cost detection on this towed platform. So the operator is being provided with a small tablet where he can see the images, and he can also see the counts of like how many cars are being detected. So it is not just like taking one car, it is actually tracking all of those cars there so that we don't do the double counting of any of those detected chords. And it can be used in like many pest-infected areas as well. Like it is very safe to operate there. Here is like a small video of our platform that we are developing. If you will see that there's a yellow cable attached to this one, which is being used for control and data transmission. However, all of those AI is being deployed onto this platform. So, so it has these 4 thrusters on all the corners of this one, which can help stabilize it and like do the control of this one. So this is the images and videos that we are actually getting in real time, and we are processing it on board. And the small video is actually the one that the vessel operator is being using to control and steer this platform. Yeah, so you can see that we can like have a like much better color, or you can say images of this area. So we have collected this data across several reefs near the Townsville and as well as the Swains area. So, yeah. So the second component is actually real-time AI for chords and scars detection, and this is the main driving thing, that how we are actually doing this thing automatically. So the as I already told that the TORD platform includes a small special sort of computer which is doing images and videos detection automatically. So the kind of analysis that we are doing on board is that it can actually see how many chords we have already detected, presence of any sort of scars, and we are focusing only on like chord scars in this scenario. And it can also calculate like what is the percentage cover for the coral in the images that it is collecting. And also the detections are being presented to the user, and they can validate them. Because some of those machine learning model, it can make some mistakes. So we have operators to validate that, okay, so the objects are actually really chords or not. So in order to develop these models, we are using data, which is like the main driving force behind this thing. So we developed a computer vision annotation tool that our marine biologists are helping us to annotate the images. If you can see in these images, we have like very 2 small chords. So our annotators are helping us to annotate these boxes initially. And then what we are doing is that we are actually training our own model. So for our, like for example, 5 minutes video, we are only taking few sample images in those videos, and those sample images are being annotated by annotators. And then for other images, we are actually using our computer vision model, machine learning model, in order to capture and detect all of those chords in remaining images. And then these remaining images are actually being provided to some expert annotator, and he can actually see that, okay, so few chords are like true positive, or they are like false positive one. So they can curate these things. And here is some results of our machine learning model. As you can see that there are like some boxes popping here for different sorts of chords as soon as it detects. And this is the kind of thing like it operates in real time. And even though you can see that the video is not very stable, but our model is still able to detect like the chords in real time here. Similarly, we developed a model for scars detection. For scars, we are actually using not just boxes, but we are also developing a semantic segmentation-based model. So in this scenario, we also provide these images to annotation experts, and they are actually labeling these areas that, okay, so these are the kind of scars they are interested in. For the scars, we also have like different sort of subcategories for them. So for example, these are like a bleaching scars or coarse scars or like drupula, these sort of ones. So however, when we develop our machine learning model, we are only focusing on coarse scars. And if you look at this image on the left-hand side is the output of our model, and on the right-hand side, you will see the ground truth that our annotators had provided us. So you will see that the scars are like much harder in order to detect and track here. However, our method is like initially providing some more false positive in this scenario. Yeah, sure. Thank you. Okay, and this machine learning model is only in proof of concept phase, but it can still provide like a very good initial estimate that, okay, so which areas are actually being detected as more scars. So the third component in our model is actually how this data workflow can be used in an overall system. So all the data and the detections are being saved in this CCIP information system, and it is being used for the reporting purposes. Secondly, that all the images that we are capturing on this onboard vessel platform, we are actually saving them later in this AIMS Reef Cloud system for any post-trip image analysis thing. And also the AI models can be refined further in the future in order to have to increase the accuracy of our systems and how we can evolve our models later on, and that can be applied as well. So finally, what are the main take-home messages from this project? First of all, is that these new technologies, especially computer vision and machine learning, has the ability to automatically detect and monitor large-scale areas, and it can actually work in a like much efficient, much better manner. Secondly, it requires like much less human expertise. They are required during image annotation, collection, sort of thing, and also just for verifying that the model is running properly or not. Second thing is that data is like very important, and then all of this sort of analysis, how many cords, how many scars, which areas are they present, they are like saved in like a proper systematic manner, and we can generate reports based on that. It also provides a better safe and efficient way to analyze large-scale areas. And also we have seen that there are like easier pathways to get like a permanent record for any kind of reefs and for future understanding. And finally, that it gives a pathway for new technologies to be taken up and used as it is. All right. So I would like to thank our partners in this project, especially AIMS and the GBRF. I also would like to thank the course control team, especially the Blue Planet Marine and Pacific Marine Group and INLOG. Thank you.
Thank you very much. Huge breakthrough. We probably have time for one really quick question. Yeah, Rena?
Thank you. Do you have any validation data in terms of what the accuracy they're responsible or anything like that for the code detection?
Yes.
So the question is for the tape. Are there any validation data available?
Yeah, thank you. So for all the trips, what we usually do is that we capture, we use like 80% of the data for training, and we always take like 20% of the data as a validation set. So our annotators are providing annotation on those images, and then we check our accuracy on those validation data sets. So However, on the validation data, usually our accuracy is like very high, but the main thing is that how this thing works in the real world, that is like the main thing. And actually AIMS can provide those numbers. I cannot actually, yeah, thank you.
Okay, thanks very much, Zeeshan.
Thank you.
So our next speaker is, I hope I pronounce it right, MD Zahidul Islam. No, no?
Emma Lawrence.
Oh, sorry. All right, that's better. Okay, we've got 3 more talks. All right, Emma Lawrence from CSIRO, a comprehensive monitoring strategy for COTS in the GBR. Thank you, Emma.
Thank you. So my story is I'm a statistician, and we don't always get tasked with the most exciting projects, unfortunately. So to be able to work in the reef space with really complex and interesting data and really important decisions to make is really exciting. Plus, I get to work with this mob who have a really diverse skill set and are providing a lot of input into how to come up with a comprehensive monitoring strategy for COTS in the Great Barrier Reef. So the last 3 talks have all been about monitoring tools or techniques that have been developed as part of the COTS Control Innovation Program. So this is about how we're all working together to bring those learnings from those projects together and come up with a comprehensive monitoring strategy. So the Reef Authority has been coordinating manual COTS control in the Great Barrier Reef Marine Park since 2010, and they've worked with partners to really innovate in this space. So they're continually improving the way that they monitor and collect surveillance data in the reef. So effective monitoring of COTS and coral really underpins the on-water delivery of the program. And it's also really integral in measuring the effectiveness of the program. They're spending lots of money. How effective is this program on its delivering its outcomes? So what we're trying to do is predict, detect and respond to outbreaks. And to be able to do that, we need a whole lot of data to be able to answer the questions about what's going on in the Great Barrier Reef. In particular, we need information on COTS size and distribution, coral cover, and the environmental factors that might be leading to outbreaks. So there's lots of existing monitoring programs in the reef, right? But all of them have a specific purpose, and no one of those, or even putting them all together, doesn't allow us to answer all of the questions that we have about the COTS, how COTS effectively operate in the reef. So here we've got a chance to consider the inclusion of more modern, potentially more accurate and also less labour-intensive survey techniques that could be used in a monitoring program. So you can see on the right here, I've taken this from the COTS control dashboard. The COTS control program collect mountains of data on a daily basis. So we're looking at how can we improve that data collection and how can we subsidise that data to answer more questions about COTS in the Great Barrier Reef. So our project has really looked at 5 key areas. So what are the monitoring objectives of the control program? What data is needed to answer those objectives? What are the monitoring tools that are most appropriate for collecting that data? Where should we be directing that effort? And what are the operating constraints and options for implementation? So we've undergone this extensive exercise to determine the range of monitoring objectives in the reef related to both management and science. We've talked to managers in science to work out when they're trying to ask questions about COTS in the reef, what data don't they have to be able to answer those questions? Now, when we went through this process, there were 27 monitoring objectives identified. Now, I've designed a monitoring program or 2 in my time, but 27, potentially competing objectives is next level, right? So it was a relief when we broke those monitoring objectives down and looked at what data did we need to answer the objectives. So basically they fall into 4 categories. One is day-to-day management. So this is the operational sort of level stuff that the COTS control program are currently collecting mountains of data. It's really short time scale at a site and reef level scale. Then there's the strategic planning for future management side of things. So that's more 6 to 24 month time scale, and it's at a higher spatial scale, so more like the reef and region sort of level. There's measuring outcomes of management, and this is longer term data, so we can see how effective is the control program being. And finally, there's science and knowledge gaps, and you can imagine the data requests from the scientists are many and varied at every scale and every possible variable. So I won't go into that too much today. So when we look at the data needs to answer each of those questions, so in the first column in this table, I've got the 4 categories of the monitoring objectives. And then across the top, I've got the data that, some of the data needs to answer those objectives. And I guess the key message here is it's consistent, right? So almost all of those monitoring objectives require COTS densities, COTS size, coral cover, coral condition, and coral type. There's various other things, but really the 2 key kickers are COTS densities and coral cover, a variety of scales. So some of these data needs are met through current COTS control program and other monitoring programs. So particularly that day-to-day management side of things is well and truly covered off by the COTS control program currently. It's more the strategic and the looking at the long-term management side of things where things get a bit more difficult because we need to look outside of the day-to-day activities. There's also programs like AIMS, LTMP, Reef Joint and Field Management Program, and the Eye on the Reef RIS that all help to add to the bigger picture about the data. Okay, so moving along, what tools are most appropriate for collecting this data? So you've just seen 3 great talks about tools that we have available, and obviously Manto is the backbone of many of the monitoring programs in the reef currently. So how do we decide which tools are best to use in the monitoring program? So the metrics differ, the ease of deployment differs, the scale differs, they have different costs. Some measure low COTS densities, some measure high COTS densities. They also really differ in accuracy and precision. So how do we decide which ones to use? What we've done is we've compared all those characteristics both qualitatively, but also we did a big comparison calibration trial. So this was carried out in March last year, and it was a beast unto itself. So it had more than 20 people on this trip. There were 7 organisations. We had the Queensland Parks and Wildlife vessel. We had the Pacific Marine Group working alongside that vessel to do manto and culling. And so we had each COTS and coral monitoring method deployed side by side with every other method. We went to 7 reefs at 16 sites at 7 reefs off Townsville. And if you see here, we managed to deploy almost every method against every other method at a site level. It wasn't all roses, so unfortunately the COTS densities were quite low in the area that we went when really we were striving for a range of densities. So that gave limitations in the data, the implications for the analysis post the data analysis. But it's still given us a really good insight into how each tool compares to each other tool. So just a really quick snapshot of what the calibration looks like. So I've just picked 2 here. On the left, we're calibrating manto against the number of COTS culled per minute. So you can see it's a positive relationship. That's excellent. But I don't know if you can see the uncertainty there. It's huge, right? So, and one of the reasons is, despite seeing many COTS on some of the culls, we saw 1 scar on manto, right? So that's pretty hard to calibrate. And it also shows a big limitation of using manta in the field. On the other hand, on the right-hand side, we've got cull versus salad. You can see the relationship is quite nice. Still a bit of uncertainty, but that's, you know, a limited number of observations, and you can see there that they're tracking quite nicely, particularly at those lower sorts of densities. So we did a similar thing for coral in a subsequent experiment. This is manta coral versus reef scan coral. So the tool that Zeeshan's been showing also calculates coral cover, and this is how they calibrate against each other. Still very noisy. That's the reef at that sort of manta site scale. there's always going to be a lot of variability. Okay, so finally moving on to where should we direct and how much effort do we need as part of monitoring. So the current effort is really concentrated on a subset of reefs. They're target and priority reefs, that says sites, actually reefs that are identified through a prioritisation process. Now, this is an extensive prioritisation process, so there's a reason these sites are being visited. They're both ecologically and economically important, and they're highly likely to have, or the most likely, given expert knowledge, to have cots and coral to be saved. So the question I come in with as a statistician is, that's awesome, but should you be monitoring outside of these target and priority sites? So how we went about answering this question was to do a bunch of simulations using COTS and coral community models, so both coconut and reef mod. And what that gave us was something that we can consider to be, you know, a form of reality. And then what we did was we threw a bunch of monitoring designs at those two, lots of simulations, to see what's the effect of, just say you chose random reefs to monitor and you varied the number of sites of those reefs. What's the effect on your ability to answer the monitoring objectives? What if you cluster those reefs to make it more logistically possible to get to them? What if you just go the priority and the target reefs that have been identified by the cull program? What if you spread evenly across the regions? Or what if you just go to the ANZ LTMP reefs? What are the implications for being able to answer those objectives? Now, unfortunately today, I don't have time to go through those simulations, but I've got lots of results if anybody wants to see them. Come and chat to me later on. What I will go through is this result here, which is what happens, how much monitoring do you need to do outside of the current cull program operations? Just say you were getting it wrong. Just say we were all wrong about where we thought COTS were outbreaking. The densities are actually far higher elsewhere, okay? So what these simulations show, left is CocoNet, on the right is reef mod. So it shows how many sites you would need to monitor to be sure that you're picking up on a COTS outbreak should they be happening in the reef. So this shows on the y-axis is the number of reefs being monitored, on the x-axis is the proportion of reefs with active outbreaks. So you'll see even if only 5% of reefs in the Great Barrier Reef have an active outbreak, if you just randomly pick 50 reefs in the Great Barrier Reef, you'd have a really high chance of picking up a reef, discovering a reef with an outbreak on it, right? So that says to me that it's a relatively low effort outside of what's currently being done to ensure that we're not missing out on things. So that's good news, right? Usually I have to go to people doing monitoring studies and say, you need to go to another 700, sites, and then they don't like me very much at all. So finally, in conclusion, there's really varied objectives of the COTS control monitoring program, but many of them require similar information. So I feel like that's a really good news story. Our approach in this monitoring strategy is to robustly build on ongoing monitoring programs, so we acknowledge that there's many of those already existing in the reef. But we also want to be able to incorporate some of these newer monitoring technologies like Reef Scan, eDNA and SALAD, where they make sense to do so. So these tools are likely to improve the effectiveness of the monitoring program by improving the data robustness, facilitating the monitoring of those lower densities that are really hard to monitor with things like man-to-toe. And it lets us get to reefs that, let's face it, there's crocodiles and you don't want to get in the water. Finally, coming up with a robust monitoring helps you decrease the uncertainty around the process of decision making, particularly in terms of those longer term sort of decisions that are being made. And the benefits include early outbreak detection, cost and resource optimisation, and improving the coordination between monitoring and management actions across the GBR. Thank you.
Thanks very much, Emma. Any questions for Emma on the floor? or online?
That's a relief.
Yeah? Oh, good.
Thank you.
Thank you. Great. So the next talk is by Zahidul Islam. Did I pronounce your name right?
Yeah.
I'm sorry. From CSIRO. Thank you.
Hello everyone, good afternoon. I am Zahidul Islam. So I'm going to present our work on improving efficiency and effectiveness of cost control using deep learning. And our team includes researchers across CSIRO with both ecologists and machine learning experts. What we are doing is we are trying to lay the foundation for next generation decision making for cost control. It's not operational yet. And our project is not a part of cost control program or cost control innovation program, but we have been working with people connected to both. Okay, so my work is, presentation is divided into 3 parts. First, I will try to convince you that pest abundance estimation across time and space is essential for effective management decision. And then I will discuss several challenges that can arise from this kind of problem and how we can address those using deep learning. And finally, I will share some of our results. As I'm sure you know, cods eat corals, and they tend to aggregate in large numbers. And during an outbreak, cods can eat up to 96% coral in about a year. And it has been estimated that cods have caused 42% of coral loss between 1985 to 2012. And most importantly, I think few talks already mentioned that cods is the only threat to the GBR that we can directly manage. So currently, the cost control is performed in voyages. So in each voyage, a set of divers is dropped into some locations, and they can cull all the cods they can find by injection. So to do this effectively, we need to know where to drop these divers, which means we need to know where the largest aggregation of cods are. Currently, Mentato has been used, as you already all heard from previous talks. So Mentato is quick, and it can effectively identify the large aggregation of cods during an outbreak. So what we are trying to do is, we are trying to estimate this number of cods without using Mentato, directly from the cull data that we have already collected. Broadly speaking, there are 2 types of decisions that's needed to make during cost control program. The one is the long-term, large-scale decision, strategic decision, and another one is short-term, on-site tactical decisions. So for the large-scale decisions, strategic decisions, we have several models that can answer strategy-related questions, but those are not effective for on-site decision-making, or if you want to know short-term where to drop the divers, their models, long-term simulation-based model cannot help us. So now I'll move to the next part, where I will show the challenges of this kind of modeling and how can we address them. So the current cost control program has a large amount of data for a pest management, ecological management program. But in terms of most deep learning problem, the data, we call them sparse, in that there are very limited repeated visits per site. So in a given day, we can only cover a few sites. For all the other sites, we don't have any information. And there are like more than 1,000 sites where we have performed Mentato before, and in a day, only few sites are covered. And as you can see from the figure at the top, each line represents the rate of repeated visit at a site. And you can see, like, for most of the sites, we only have few visits. And for very few sites, we have more visits. And the figure at the top shows that our data is also really highly zero-inflated data. So for most visits, where we counted some cods during Mentato, the detection, they are able to detect very few number of cods. And this is an important problem for machine learning because they struggle with this kind of sparse data. And also, this is a problem for the traditional statistical model. So they can't work with this kind of sparse and zero inflated data for regression. So here we tackle these challenges using advanced deep learning model. So to aid the management, the whole process can be described by these 4 steps. So we develop a machine learning based model. We train it using the call data that we have already collected. And then we use that model to make abundance estimation. And this cod management team, they can take this estimation and find the largest aggregation of cods and where to deploy the control actions. So there are mainly 4 challenges that can arise in this kind of problem. First of all, I already said, sparse observations. We want to model the temporal dependency as well as spatial dependency, and we want to tackle the zero inflation in the data. So to do this, we have this model. Our model has several components. First of all, to deal with the sparse observations, we have an input encoder, which is a neural network. All of these components are basically different types of neural networks. So to tackle the sparse observations, we have input encoder and a spatial encoder, and to model the temporal dependency, we have a temporal encoder. So that's kind of a recurrent neural network. And to model the spatial dependency, we have a graph neural network. And to deal with the zero inflated count data, we have the output of our model as a zero inflated Poisson distribution. And we estimate the parameters of this distribution using this model from the data. Okay, so now I will discuss some results that we have got from our model. So from a management perspective, we want, so lot of the good decision depends on the data and the good analysis of the data. So, which means that we want our model to be accurate and most importantly, precise. Mantato is quick, but we know that it's not very accurate. So if you think about it statistically, Mantato is not accurate or not precise. But our model is more accurate and precise than Mantato. So in the figure on the left, we see that the call data, actual call data is plotted as black line, and the green is our model's prediction, and the red is showing the Mantato data. So Mantato is good at predicting the outbreaks that you can see like the few bumps in the red lines. But our model is more detailed and more accurate, similar to the call data. And this is important because using this recommendation, using our more accurate model, we see that is shown on the figure on the right side, that in a year, using the same resources as Mantato, we could have removed 33% of more cards using the recommendation generated by our model. Okay, so another very attractive attributes of our model is able to predict with very few observations on the site. So here I showed 2 examples. The first one on the left is a site number 4 from Great Barrier Reef, and as you can see, like the marked dot, the actual number of cods, before that, we only have 2 observations, which are not very high. But from our model, we see that our model is predicting that in future, we are going to get a large number of cods, which is shown by the green line here. And it does it using the spatial correlation it learns from the nearby sites. And in another example, in the Lady Elliot Island reef, site number 5, we see that we only have 1 observations beforehand, and we don't know about what's going to be in the future. And our model has correctly predicted there is some high number of COTS activity that's going to happen. And it only used 1 of the observations before, and the observation that is available at the nearby sites. Of course, our model is not perfect. It sometimes do overestimations that shown in the red arrow, but overall, our model is better than the existing models we have for tactical on-site decision making. And one of the limitations of machine learning is they work like a black box, and we don't know how they are making these predictions, which is really problematic if we want to integrate this in government decision making. So what we did is we did a bit of work to pull out some of more additional information to explain why our model is doing, why our model is performing this well. And we generated few graphs to do that. And one of the, like I mentioned, our model uses a graph neural network, which is like learning a graph from the course data that we have already collected. And here we pull out some of the graphs that showing that what our model has already learned, what kind of relationship it learned from the data in terms of special correlations. So first figure here on the, that is marked at B on the left is showing that our model is using information from the nearby sites within the same reef. And also our model is also learning some of the information from the nearby reefs as well, that is shown on the figure on the right. So the problem is we don't know the ecological process or how this model is learning. We know how it learns, but we don't know the underlying ecological process that shows this kind of relationship. But this is what we infer from the data. And we also plotted the temporal and spatial scale that is our model using for inferring this kind of spatio-temporal relationship. So the y-axis shows, the x-axis shows the time difference in days, and y-axis shows the distance between the sites. And we plotted this graph using gradients of the model. So this kind of showing the sensitivity of our model in terms of temporal and spatial distances. So the contours here we see that, so our model is mostly sensitive to observations which are within 15 days time period, and the sites which are like of 1200 meters apart. So this kind of intuitive and matches with the real phenomena, because we know that a course at a site is mostly dependent on the nearby sites, course at the nearby sites. And also each voyage kind of lasts for 14 days, so it's showing that our model is depending on 2 consecutive visits or voyages. So this kind of confirms that our model is basing its prediction on the actual phenomenon of the data collection. So I'm running out of time, so this is the take-away message. So we can make good predictions with the CAL data only without needing a metadata. And using these predictions, we can make better decisions in terms of cost control program. And this kind of sparsity on observations is also present in all kinds of ecological modeling as well. So we assume that our model is also going to work for those cases as well. Thank you.
Thanks very much, Zahid. Really interesting. Any questions from the floor? Yes?
Yeah, that's correct. So currently our model can only work on the sites already we have data for. So even if you have only one data, that is enough for the model to make predictions, because that data is needed because we want to know the location of the site so that we can infer the graph from that location and how many these other sites are connected to that site. We can learn from the data. So that's why we need at least we need one data at a site.
Any questions from the... If not, thanks very much, Said.
Thank you.
And last speaker of this session is Maria Byrne, and looking forward to your talk. Thank you.
Thanks very much, Katharina. Thanks, everyone, for coming, and good afternoon. My little story, very short, is yes, Jacques Cousteau had a lot to do with me becoming a marine biologist, but I wasn't in the Mediterranean. I was in 5-degree water. I learned to scuba dive in Ireland with a wetsuit when I was in university. 5 degrees in the winter, 10 degrees in the summer, but hot whiskies were actually quite good to get over the hypothermia that you experienced every single dive. Anyway, so today I'd talk about the research that we've done on the juveniles. And this is just the team of people that's been working for us, and lots of people to think, like the COTS control and all the supply train. And I'd like to acknowledge the traditional owners of the Great Barrier Reef and its catchments as the First Nations people holding the dreams and traditions of this beautiful area. But also the Gadigal people of the Eora Nation is where I do my work, because I haven't been working on country, because we get our COTS shipped to Sydney. So we do all our work on COTS in Sydney. All right. So COTS life cycle, I won't say very much because most of you know about it. It spawns, has a larva. Now, what we worked on was this stage here, the transition between the very, very early juvenile and this pink later stage juveniles, which is the herbivorous. They're pink because they eat coralline algae, and they get the pigment of their food. And then, of course, then you've got the coral eating stage, is where we have stopped our work. So these animals are, our work sort of started off with the hidden army hypothesis. I had a student about 12 years ago doing ocean acidification and COTS, and she had some leftover juveniles, and so we didn't know what to do with them, so we just threw them. Looked at them after 2 years, they're still there. Looked at them after 3 years, they're still there. I says, oh, right, I know what's happening. So let's have a look at this possible hidden army hypothesis. So then another student came along, Dion Deeker, and then she did this work. And yes, indeed, they're there. They are very hard to detect. And so obviously they're very, very hard to study. Jennifer Wilms, a shout out to her, because she did a heroic job of finding these darn things on the reef. Pretty amazing. So the aims of this work was to look at the influence of waterborne cues, the behavior of these juveniles, to their various cues that they would get in the environment. Crossed-house coral and alga, that was a CCA covered with rubble. My suitcase was full of rubble that I brought home from Montreal Island, so it was the real thing, and some adult conspecifics that you got courtesy of the COTS control divers, and various combinations. Our hypotheses were that the juveniles would be able to identify their food source, the CCA, as also a nursery habitat as well. But when it came to the adult conspecifics, we weren't sure. Would it be a signpost to go there because that's where the coral is? Or, as we found it with Marthasteres glacialis in Ireland, was that maybe they would not be attracted to the adults. So let's just say sort of indication of a density-dependent mechanism happening. So this was the setup. I won't go into any great detail. It was just the header tanks. This is the flow chambers, and a shout out to Mike Kingsford, because he had put juvenile fish into the same thing that we put juvenile COTS in. It was a really good array that we finally decided that works for these juveniles, and we presented the cues in flow through, the Y maze, and then we characterized whether they went up or down, which direction they went. And we did our experiments under red light. Initially, we did them using police body cams in the complete dark. Thank God we didn't have to keep doing that. And then eventually we got the array working really well, and then we used time-lapse photography and tracked them. And the data analysis there, just for your completeness. So just don't worry about this figure on your left at the moment. You're going to see it again. So we had 4 cues. Types, we had just plain old filtered seawater, 1 micron UV filtered seawater, try to avoid any bugs that might be in Sydney Harbour. And we've got the coral and CCA and adult COTS, and a various combination of these cues, and how would they respond. Just filtered seawater only. This is sort of expected Some moved upstream, some didn't move at all, and the downstream movement was significantly different, but most of the cots either moved upstream or into no movement, and sea stars tend to move upstream, so that wasn't too much of a surprise. These are the choice responses, and again, you're going to see these again, but we have, if you look at this, is that you've got CCA rubble, blue is just water. Oh God, my sight is horrible. So we've got the pink is the CCA, the blue is filtered seawater. The direction is that either downstream or upstream, and you'll be seeing more of these choices now, and I'll go over the data with you. So the choices with single cues. So this was the run with just single cues, and so you have here CCA, the cots, and the coral. The color seems to have changed in this. Anyway, so they went for the yellow, and is the habitat, either the CCA or the coral, and the red is the avoidance. So this was our first indication that the juvenile cots actually avoided the cues from the adults. And then you've got with the two choice, which then made things a bit more complicated. And that's the two choices in this one, is that you've got coral attraction. They preferred coral to CCA. This was at the stage when they were able to eat coral. But then when we got to the combination of cots and coral, the no movement was strongest. And when we had cots and CCA, we had no significant result. Essentially, when we saw the juveniles in the presence of an adult, regardless of where the nursery CCA covered rubble was there or where the coral was there, the juveniles meandered. They weren't sure what to do. It was definitely not a black and white response as we saw with a single cue. So to try and tease this out, then we took our movies, so we filmed them, and we tracked the behavioral responses. So we had, just tracking the responses. This is these squiggle lines. We literally tracked every single individual. And so first of all, we'll got to the single cues. So when the rubble was by itself, CCA covered rubble, this was the most direct path. The juveniles mostly went straight to the CCA rubble arm of the Y maze. It was the shortest distance moved and the fastest movement. So they could definitely, and remember, these juveniles have never seen any of these cues in their life. So this is a first, they're naive. The tracking response to corals, that was interesting. Again, they were attracted to corals, and it was a direct path, the short distance, but it took the longest time for them to make a choice. They were slow. And it kind of indicated some sort of cautious attraction, and maybe that's warranted because, as people have seen and various people have published, the transition to being a coral predator is a bit hazardous for cots because they can get stung and killed by the coral tentacles. But they're attracted nonetheless, but a bit more cautiously. And I'll just say, I should really explain what all these axes are. That's speed of movement, time to make a choice, and distance moved, and we got all that information from the movies. The tracking response to cots, well, they really meandered. Most of them went downstream, and the rest of them kind of went up into the filtered seawater arm. And this was the greatest distance moved. They were really quite indecisive. It took them the longest time to make a choice, and their choice was away from the cot, away from the adult source of the adult cue. So what we find out from this work is that the herbivorous juvenile cots have strong sensory abilities. They can navigate a distance, navigate towards cues coming from a distance that they have never seen before. So this is an innate response of these juveniles. They move directly towards cues from CCA-covered rubble, and that indicates a strong attraction to the nursery habitat. In other words, they find home quickly. They had a positive response to coral, but it was a bit more tentative, and maybe that hesitancy with respect to the dangers of the juveniles in their transition to the coral eating stage. When it comes to the adults, they moved away from the cues of the adults, and the adult avoidance may indicate what we might see in here, and we've seen in other species of sea stars: density-dependent recruitment, a population feedback mechanism, and that's whereby they avoid competition from the adults. If the adults are there, they don't emerge. And this really does support that juveniles in waiting hypothesis, and that the juveniles can accumulate in the reef infrastructure over multiple years and transition to coralivory, transition to become coral predators as the opportunity arises, maybe after a COTS outbreak has gone and there's still food around. There's more work on that to tease that one out. And really what the management implications of this are that the COTS control programs, when they remove the adults, do they release the juveniles from competition? So that causing the juveniles then to switch to a coral diet, that hidden army emerging. And because we know that some reefs on the Great Barrier Reef, even Moran said that 20 or 30 years ago, that it takes a long, when you remove the adult COTS, all of a sudden there's more COTS there again. Just when you think you've got them all, you come back, there's more again. So that could be that more again may be the juveniles emerging once they've been released from competition from the adults, that hidden periodic emergence of the hidden army. And COTS, as Charles Birkeland said, has a lot of Faustian traits, and this is one of the Faustian traits of COTS. It's really an inconvenient truth, really, that they have huge plasticity. That's one of the reasons why they're so successful. They have stress resilience. They're really tough. Both the juveniles are quite tough, and they might be in the reef infrastructure, and once the availability of, when the coral prey is low, and then such as following a bleaching event or a cyclone or a COTS outbreak has gone. So when there's low coral cover, the juveniles are still there, but in the reef infrastructure as herbivores. So the implications for coral reefs faced with climate change is in this scenario is pretty dire, really, is that if in a warming world, because we do know the juvenile waiting stage COTS are far more resilient to heat waves than the corals. 12-degree heating weeks did not knock them back at all. The coral was all dead, but the COTS were all quite tough. They don't mind heating. To a certain extent. They didn't like 36 degrees, but neither does the coral. So the warming world residing in the rubble may benefit from an increase in the extent of this horrible. So this is an insidious feedback. You've got bleaching, as we're seeing now on the Great Barrier Reef, and I tell you, one tree island was just smashed in heron. The rubble from the bleaching, that rubble collapses. It gets covered in algae and CCA, and that's the food of the COTS. So now you get an insidious feedback whereby coral bleaching kills COTS, creates more rubble. Rubble gets covered with coral and algae, and that's happy days if you're a juvenile COT. That's not happy days if you want to keep a reef happy. This is the scenario from that paper, waiting stage juveniles. If you see them here in the little coral, this is this little cartoon that my student Matt made. They're there in the rubble, and this is a normal reef. Then the reef bleaches, and then it gets covered with algae, but the juveniles are still there. The reef collapses, way more coral, rubble, a lot more food. And eventually you'll get something like an acropora recovering very quickly and growing, but the juveniles there are in waiting. So it's an interesting scenario, but I think it's a scenario that we really need to take on board and considering in our management models. And that's it. And I'd like to thank everyone that's involved in that. This is Monique, who did all the work in the dark. She's a very tall woman, and the crew. And I'd like to thank the the Great Barrier Reef Foundation and the CSIP guys, COTS control guys that got the COTS first in the first place. Thanks very much for listening, everyone. Oh, bye, Steve.
The issue with releasing the juveniles, do you know how big or how adult an adult needs to be before the juveniles are wanting to avoid them? So does a 15 centimetre adult still have the same effect on the juveniles and making them avoid? I'm just thinking because when we control, we usually, the 15 centimetre adults we're not really getting. So I was just wondering if that's enough to keep the, would that be enough to keep the pressure on them?
That's a very good question, but I would suggest that the chemistry of a corallivorous cod, whether it's this size or this size, is probably quite similar. The only difference between a very small cod that eats coral and a big cod that eats coral is probably the gonad chemistry, the reproductive chemistry. So I suspect not. I suspect they're just detecting competitors out there.
Yeah, so I'm just, in that case, it could be, if we are releasing the pressure, if we're focusing on the larger individuals, we still might get the competition by leaving the smaller adults there.
Yeah. You have to take everything off.
Yeah.
Yeah, thanks, Maria. That was a good talk. I just wanted to comment, or maybe hear your comment. If the juvenile in waiting is true, it's obviously, because to be honest, not all of us have supported culling all the time, because we were worried that it would lead to what we call the Okinawa situation, right? That it's a continuous outbreak, we're culling and new cods are coming up faster and faster. So if your juvenile in waiting was true, that would be one mechanism to create this and would probably, could be taken as an argument against culling cods?
Yes.
Thanks.
Yes. I think you've really got to understand what the feedback is between the juveniles, and there may be a critical number. Like, for instance, Marthasterius glacialis in Lochine in west of Ireland. Those big Marthasterius, and they're really big sea stars. Their density did not change in 20 years, but the juveniles were always there in the waiting. So all we could figure is as one of the adults popped off, another juvenile would pop in, but it was stable for 20 years in 3 different habitats. The recruitment habitat, the intermediate habitat, and the adult habitat. Something happened similarly with Pisaster or Cratius in west coast of North America, and Mary Sue found that one.
Well, sea cucumbers do that by asexual reproduction.
Yeah.
What this is for, but they keep very stable populations just by replacing all mortality with asexual.
So really sorry, we could go on forever with talking about echinoderms, but the interesting thing is it does run through echinoderms, and I think normally, yes, echinoderms will not be so silly to eat themselves out of house and home, but something's gone wrong with the barrier reef, and it could have been overfishing.
So you can work 3 questions from the spot, and then the plenary will start in 6 minutes over the
Sorry.
Okay, I'll try to quick. Maria, another question about this release hypothesis. I just wonder what happens when you take into account space, in the sense that, you know, if you've got areas of rubble versus areas of live coral, is it going to apply so much in the sense that, you know, the rubble is great habitat for the juveniles, but maybe there doesn't tend to be a lot of adults around because they'd prefer to be on the live coral?
Yeah, no, that's a good, those juveniles that are in the rubble that don't have adults around, yeah, they're probably not getting that communication. But once they grow up, they're going to have an imperative to grow. So they will biologically want to move to a coral-eating space, adults or not, because they want to grow up and want to have children. So the thing is, that's what Zan showed in Fiji. It took 2 years for the juveniles to move from the reef flat to the reef crest. And they went over 50 to 100 meters, those juveniles moved.
Okay, thanks, Maria. I think we could talk about cods for the next 50 years. I would like to again thank all the speakers this afternoon for really, we're coming so much further with the types of monitoring, how we can deal with Crown-of-Thorns, the types of understanding of the basic biology. I mean, for heaven's sake, we think we've Crown-of-Thorns for a while. So much to learn. So, thank you, everybody. And the next speaker is Mark Gibbs in the urgent room and the next plenary. Then an afternoon tea break. Then a panel discussion, and then the reception. Thank you.